Hello students, welcome to Vedant NEET English. This is 𝐂𝐡𝐞𝐤 your biology master teacher. I hope everyone is safe and sound and doing extremely well in life. Now students, in this class of Bio Club, we'll be doing two chapters. I'll be doing Living World, Gopika ma'am will be doing the next chapter, that is Biological Classification. Okay, so without wasting any time, quickly let me know in the chat right now if I'm audible and visible to each and every one of you. Quickly let me know in the chat right now. Quickly, every single one of you. If my voice is audible, check, sound check. And if the board is visible to all of you. Quickly let me know in the chat, students. Quickly let me know. And good evening to each and every one of you. Good evening, good evening, good evening. Now let's ask a very controversial question. Let's start the class with a very controversial question with go. Right? The controversial question is very simple. What is life? If someone usually asks, right, how is life going? Do you ask your friends? Like I usually ask my friends every single time I meet them, how is life going? How is your life? How is your life? But what is this life? What is life, students? Now there are two perspectives to it. First perspective is religious. Like full my chakra points are open. I am very religiously active. That is life. The other example, the other meaning of life is science-based. And that is what we are going to understand, the science-based explanation of what is exactly life. Now students, can anyone in the chat tell me what is life? Anyone? Can anyone in the chat tell me what is life? What is life? If you ask me, I will tell you life is nothing but lot of molecules coming together. I repeat again, life is nothing but a lot of biomolecules coming together, interacting, and the chemical reaction which comes out, that is called as life. So what is life? Life is nothing but chemical reactions. Okay. Now students, for example, whenever there is a birth of a child, right, the child will grow. Once the child is growing, will he learn something new? He will learn something new. Will he adapt to certain conditions? Yes, there is adaptation is there. Finally, the organism will die. In this entire phase, there is a phase of even reproduction also. Reproduction. So there are multiple things a organism goes under, right? In its lifetime, it could be me, it could be you. All of us go under a lot of different changes during our life. We grow old, we learn something new. For example, if you sit in my class today, you will understand this entire chapter. That is what you're learning from today's class, right? But students, apart from this growth, adaptation, sustainability, there are five main things, or I will call it as a basic features of life. Those basic features of life are nothing but consciousness, first one. There is metabolism, there is reproduction, cellular organization, and growth. These are the five different types of features of living organism. And this is what is given in your NCERT also. So let's focus on these five features and let's classify them based on a few parameters. So students, if you are able to understand till now, I want everyone to quickly smash that like button right now. Every single one of you, quickly smash that like button right now and we'll go to the next concept. Okay, let's go to the next concept. Now students, we understood that there is consciousness, metabolism, reproduction, cellular organization and growth. Five different basic features, right? All of you agree? Five different basic features. Now what you need to understand here is, we will classify these five different features into two separate categories. Understand, right? Basic characters, or these parameters can be classified into two separate categories. The first one is called as fundamental. It is called as fundamental. And the second one is called as defining. Now, first of all, I want you to understand what is the difference between fundamental and defining. Understand the difference. After understanding the difference, next step is classification, where I'll keep reproduction somewhere here, I will keep cellular organization here, I will keep metabolism here, I will keep growth here, right? The basic classification will come later on, but first of all, I want you to understand what is the meaning of fundamental and what is the meaning of defining. Ready, all of you? Ready? Okay, listen to me, all of you, listen to me. What is the meaning of fundamental? For example, plants are green in color. Plants are green in color. Is it fundamental or defining? Tell me in the chat now. Plants are green in color. You're like, sir, plants are green in color. It's true. It is true. Plants are green in color. Or I will say, leaves are green in color. Now students, have you ever observed any other color of a leaf? Have you ever observed a white color leaf? Have you ever observed a yellow color leaf? Have you ever observed a orange color leaf? Have you observed in your life? Have you observed that? So if I make a statement, right now, leaf are green in color, are there any exception to it now? Are there any exception? Yes. Because a leaf can be green, it can be yellow, it can be orange, it can be light yellowishly orange. There are multiple possibilities are there. It is there, possibilities are there. So when there are exceptions, are involved, if there are exceptions, then a feature or a parameter is fundamental. For example, color of a leaf is fundamental. Why? Because there is an exception there. Okay. Now, I'll ask you one more thing. All the plants have chlorophyll. Now again, every single plant belonging to plant kingdom, will have chlorophyll. Yes, it is a universal pigment in plants. All the way from the algae to bryophyte, pteridophytes, gymnosperms, angiosperms. Every single classification of a plant will have chlorophyll. There is no exception. So henceforth, such a feature which does not have any exception, that is, there is no exception, that becomes defining. So a fundamental feature will have exception, a defining feature will have no exception at all. So based on this primary information for all of you, can we go to the first type of feature now? Can we start all of you? Can we start the first feature now? All of you? The first one? Yes. The Hulk. Okay. The first one is called as growth. The first feature is called as growth. Now, I'll give you a simple example. Tell me if it is growth or not. Every growing thing consumes energy. True or false? Every growing thing in this world consumes energy. And it is living. Everything which grows consumes energy and it is living. True or false? Could you tell me in the chat? For example, let's say I go outside my room, I eat biscuits, I'm consuming energy from the biscuits and I'm growing. Let's take one person from example, let's take Karthiki or Meghana, they're eating some noodles nicely, they're consuming energy and they're growing. Is it always true? Not really. Why? For example, fire. Does fire also consume energy, students? Tell me. Does the fire also consume energy? Yes, sir. But is it living? No, it is not living. So, not true. Okay. So, before we go to the actual growth, entire growth can be classified. What is growth? Growth is basically increase in cell number or increase in mass. So, increase in cell number and increase in mass, these are called as twin. They are called as twins. Twin characters of growth. So what is growth, students? Increase in cell number, increase in body mass. I'll give you one more simple example. Tell me, students, which was the first cell in the human body? The first cell in the human body, which was a diploid cell called a zygote. Now, from one zygote, one small kuti, you can't even see it with naked eye. From one small zygote, you became a multicellular organism like you and me right now. Right? Why is it happening? We are showing growth. What exactly happened there? There was increase in cell size, increase in cell number. From one zygote to multicellular body, increase in cell number. That is growth. That is growth, students. That is growth. Now, one thing which needs to be taken care here is, right, in the entire growth, anabolism should be more than catabolism. Does it make sense? Does it make sense to all of you? Why? But imagine you're trying to make something, but before you make it, it breaks down, you will never grow. So anabolism has to be more than catabolism in the case of growth. Okay. Now, students, one question to all of you for you to make your brain think more. Okay. Now, in the case of unicellular organisms, the mode of reproduction is generally binary fission. It is generally binary fission in the case of unicellular organisms. It is generally binary fission. Now, what is so special about it, students? What is so special about it? The speciality about unicellular organisms is that here growth and reproduction are same to same. If a cell is dividing, it is growing. If it is growing, it means it is reproducing. So here reproduction is equal to growth. If an organism grows, it reproduces. If it reproduces, it grows also. Now one question arises to all of you. Can anyone tell me this bacteria belongs to which kingdom? Can anyone tell me in the chat quickly? Bacteria belongs to which kingdom? Quickly, all of you. Can anyone tell me bacteria belongs to which kingdom? You'll tell me kingdom Monera. Kingdom Monera is the thing. Okay. Now, this one more question is here, right? Why do you think primitive unicellular prefers this mode of reproduction? Little out of NCERT, but it makes you think, right? It makes you think. Like we humans, we have sexual reproduction. Now, what is so great about unisexual reproduction in the case of bacteria? What is so special? The answer is very simple. Because this entire method is very simple and efficient. No, you don't even require a partner here. Not require a partner. Less genetic variation and very easily being done. Less energy requirement. Okay. Now, students, let's take to one step ahead now. There are two types of growth here. Something called as extrinsic growth and intrinsic growth. Very basic, understand very carefully. What is the meaning of extrinsic growth right now? Extrinsic growth. Now, students, tell me what is the meaning of X? X means outside or inside? Use your simple brain cell and tell me X means outside or inside? Quickly tell me in the chat, all of you. X means outside. For example, this is a mountain. On top of this mountain, on the external, that is outside of the mountain or a hill, there will be accumulation of sand or snow. Let's take snow as example here. Snow here. Snow is accumulating. So it is growth from the outside. Such a growth from the outside is called as extrinsic growth. Now, students, tell me what is the meaning of in? In means inside or outside again? In means inside. So growth from inside is called as intrinsic growth. Now I'll ask you one more question to all of you. In the case of living organisms like me and you and all of us, do we show extrinsic or do we show intrinsic? Quickly tell me in the chat, all of you. Do we show extrinsic or do we show intrinsic growth? Obviously, you cannot see anything growing from outside here. Nothing is growing from outside. Everything is from the inside. So in the case of, let's say, humans as a simple example, we show intrinsic growth. We show intrinsic growth. Done. Extrinsic means outside, intrinsic means inside. Done. Simple. Now, growth can again be divided into definite and indefinite growth. Indefinite growth. Now, all of you look at this simple cute kitten here. This cute kitten is eventually growing into adult cat. Okay. Now, students, tell me, after 18 months, does this cat eventually become the size of a house, size of a building, size of the planet? No. Does the size become constant after some time here? The size is constant after some time? Constant. Take humans for example. Humans stop growing, boys generally stop growing after, let's say, 22 to 23 years maximum. Do we stop growing after that? Yes, we do. I'm talking about vertical growth, right? So growth is limited to a certain age. That is called as definite. That is, growth is always limited. Growth is always limited here. Limited growth. Now, students, have you noticed plants are neighbors? Plants will keep on growing throughout their life. Like I have this plant I have seen which was there in my school. When I was there, it was, let's say, around few meters wide. But when last time I visited my school, it was so huge that I could barely recognize it. It's been more than 10 years, obviously, but a plant or a tree keeps on growing. That is unlimited growth. Unlimited growth. Such growth which is unlimited is called as indefinite growth. It is called as indefinite growth. Now, can anyone tell me in the chat what is the secret behind the success here? Can anyone tell me what is the secret sauce they're using? What is the sauce here? What is the secret sauce here? Indefinite growth. Anyone? They have an amazing power, no? They have a power of dividing, which we do not have. They have a special type of tissue called as meristematic tissue. How many of you are aware? Right, they have something called as meristem, primary meristem, secondary meristem. All of you remember, no? Plant, right? Anatomy. So that was your growth. Now, students, a question to all of you: is growth a defining feature or of a living being, or is it fundamental? If you know the answer, tell me in the chat quickly right now. Is growth a fundamental or a defining? In the chat. Now, students, did we observe exception to growth? Now, see, this is also growth. Growth from the outside, growth from the outside, growth from the outside. So all of these are exception to the growth. That is, non-living things also showing growth. Non-living things also showing growth. So I will call growth. I will place growth under fundamental. So I'm placing growth here. I'm placing growth here. Done. Next one is called as, next feature is called as reproduction. What is reproduction? All of you know. Reproduction is when an organism gives birth to a young one which is similar to itself or different from itself. Students, tell me in the chat. When an organism is giving birth to the young one, does it look similar to the parent or dissimilar to the parent? Similar to the parent. You will never see in nature an insect is giving birth to a hippopotamus. You will never see in nature an insect is giving birth to a hippopotamus. Not possible. Okay, students. So do you want to know something funny? Every single time hippopotamus, this comes to my mind. Okay. Last time I went to zoo. Okay. My mother and I, all of us went to the zoo, and for some reason, my mom did not know it is called as hippopotamus. She kept on calling it a happypotamus, and I was wondering what is happypotamus for the whole time. So every single time I think of hippopotamus, I think of happypotamus now. I don't know why. So happypotamus, right? So insect will never give birth to a happypotamus. So stupid and made funny. Okay. So that is called as reproduction. Okay. So one organism is giving birth to a young one which is similar to itself. Similar. Okay. So there is nothing called as other thing will not happen. That, like a dog will give birth to a puppy. A dog will give birth to a puppy. A cat will give birth to a kitten. A human will give birth to a human. Okay. Now, students, tell me in the chat quickly. all of these examples are for sexual reproduction. All of these examples are for sexual reproduction, right? Sexual reproduction. Now, do you see clones in sexual reproduction? Do you see clones here? That is a parent to offspring. A parent to offspring. Is it exact copy point to point? No. We do not produce clones, obviously. Clones are not produced in sexual reproduction. Clones are not produced. There will be certain variation. There will be certain variations are going to be there. Okay. Clones are not produced here. Okay. So in sexual reproduction, offsprings vary genetically from the parent. Asexual reproduction, offsprings identical to the parents, that is clones. So clones are produced in the case of asexual reproduction. Asexual reproduction, clones are produced. Okay, I hope that is clear to all of you. Okay, now in the case of asexual reproduction, quickly tell me in the chat how many parents are involved. How many parents are involved in asexual reproduction? I want stuff, rapid answers. This is slow pace. All of you are very slow today. All of you are very slow. I want everyone to be in high energy, fast paced answer. I want to see quick answers there in the chat. Tell me, in the case of asexual reproduction, do you find single parent or multiple parents? It's going to be uniparental. It's going to be uniparental in nature. Uniparental. Now, what about in the case of sexual reproduction? Do you find a single parent or do you find two parents? Tell me. It is going to be both. It is going to be both actually. Not just two, it is going to be both. Now, some of you might be wondering, sir, how both or how only one? In sexual reproduction, we generally have two parents, no? One male and female parent. But how both here, students? How one can also be here? Like both make sense. Biparental makes sense. One male and female. But how does the uniparental also make sense here? Let me tell you. If you're my student, you should be able to tell. If you're my student, you should be able to tell right now. What is the answer? Very simple. All of you know a famous flower called as hibiscus flower. All of these are called as bisexual flowers. Bisexual flowers have both male and female reproductive structures in a same flower. Stamen and pistil are present in same flower. Flower is one, organism is one. So one organism has both the sex organs. Sexual reproduction is possible. Hermaphrodite. Yes or no? Yes or no? Yes. Simple. Okay. Now students, the next thing which I have to tell you is a few examples here and there. If you know the answer, tell me in the chat quickly right now. The mode of reproduction in other organisms. Mode of reproduction. Mode of reproduction in other organisms. In other organisms. Okay, other organisms. Let's take the first one as let's say in case of fungi. Students, tell me. In the case of fungi, which type of asexual mode of reproduction do you notice? In the case of fungi, tell me. In the case of fungi, kingdom fungi, do you remember basidiospores? Yes, you remember. Do you remember aplanospores? You remember. Do you remember planospores? You remember, right? So in the case of kingdom fungi, we generally notice spore formation. Spore formation. We also find fragmentation also here actually. We also find fragmentation also here. Okay. Fragmentation also here. Okay. Next one. If you know the answer, tell me. What about in the case of yeast? Yeast or hydra? In the case of yeast or hydra, do you remember students? This diagram like this. What is this structure? What do you call this? We call this as budding. We call this budding. Okay, budding can be seen here. Now, what about in the case of, let's say, planaria, the immortal being? Students, there's a story, remember, Rakta Beach, right? You cut every single time this Rakta Beach demon, one drop blood falls somewhere. From that drop of blood, one more organism, one more Rakta Bij will arise from there. How many of you know the story of Rakta Bij? You remember Rakta Bij story? Like one drop of blood falls somewhere, one more organism will grow there. Like one more Rakta Bij will come. This is Rakta Bij. Same Rakta Bij. You cut one plant, one more part will arise from there, right? Basically what happens here is regeneration. I wish I had this ability, students. I wish I had this ability. Super fascinating. Regeneration. So cool this is. So cool. Okay. Next one. Let's say in algae, students. Algae. Which asexual mode, students? Fragmentation is there. Yes, there is fragmentation. And also spore formation in few cases. Spore formation in the cases. Okay. That's it. I think as much as remember. As much as enough. Let's talk about even mosses. Last one. In the case of mosses, we find something called as protonema. Protonema formation. Okay, protonema formation. This is clear to all of you. Rakta Bij you should know, students. Hindu mythology. Here and there. Okay. Now question to all of you. If you know the answer, tell me in the chat right now quickly. If you know the answer, tell me in the chat right now. All of you. Can all living things reproduce? Or are there certain organisms which cannot reproduce? Ayo papa. Sad. Tell me, students. Can all the living organisms reproduce? Or are there any exceptions? Are there any certain organisms which cannot reproduce at all? They are not capable of reproduction. Yes, there are. Yes, there are, students. Yes, there are. Infertile couples, male worker bees, drones, right? Sterile worker bees cannot, mule, liger, right? All of these artificially created hybrids of different animals are all sterile in nature. All sterile in nature. They cannot reproduce. So, if they cannot reproduce, can I call this entire slide as exception to reproduction? Yes, it is exception. So, if there are exception to reproduction, can I call reproduction as fundamental or defining? Answer in the chat. So, I will keep reproduction as fundamental or defining. Here, defining or fundamental? Fundamental. Reproduction. Reproduction done as fundamental. Next one. Next one is called as metabolism. Metabolism is very simple. Basically, sum total of all the reactions happening in your body. It could be glycolysis, Krebs cycle, electron transport chain. It could be breakdown of fatty acids, breakdown of amino acids, deamination reactions in your body. Entire, all the catabolic and anabolic reactions happening in the body of an organism is called as metabolism. It could be glycolysis, 10 steps breakdown, Krebs cycle. In the case of plant system, photosynthesis is an anabolic process. Respiration, entire thing is catabolic in nature, right? So, all of these reactions are called as metabolism. And let me tell you, all of the organisms in the world, living organisms in the world will show metabolism because they need to produce energy. Without energy, an organism cannot survive, and henceforth, respiration is must. Respiration is must. And henceforth, metabolism is a defining feature. Metabolism is a defining feature, students. Okay. Not simply in living things. It can be demonstrated outside the body in a cell-free system. It can be demonstrated. That is, all of these living reactions can happen in test tube also. They can be. But reactions are living reactions. They are living reactions. Okay. So all living organisms are made up of chemicals. I told you in the starting. What is life? Life is basically chemicals, chemical reactions. Right? All of these chemicals undergo several reactions in our body. These reactions are collectively called as metabolism. Called as metabolism, students. Called as metabolism. So where do I place metabolism? Students, quickly tell me in the chat. Where do I place metabolism? I will place metabolism under defining. Metabolism. Done? Done. Now, students, how many of you watched the movie called as Finding Nemo? How many of you watched the movie called as Finding Nemo? This right here is Nemo, okay? Nemo is also called as a clownfish. I'll take one example right now. Nemo is called as clownfish. Okay, clownfish. Now, if you cut this clownfish, RIP, okay, RIP, right? This is the clownfish organism I'm taking my test subject. If I cut it open, and I will notice that this entire organism is made up of different types of tissue. A group of cells is present in a tissue, right? If you focus, you'll find cells. If you focus on cells, you will find different organelles. That is, you will notice that every living organism, it could be me. If I cut open myself, and I'll notice that I am made up of different type of tissues, different type of cells. If you cut open a tree, you will notice different types of tissues, parenchyma, collenchyma, sclerenchyma. You will find xylem, phloem. That is basically called as cellular organization. So every living organism will show cellular organization. Okay, will show cellular organization. Organelles interact with each other to form the proper function of cell. Okay, due to the presence of organization, a living body is called as an organism. Okay, so there are no exception here. There are no exceptions to cellular organization. Every single organism will show cellular organization. Cellular organization. Now students, how many times it has happened to you that rasam is kept in front of you? How many times it has happened to you? This example is foreign example. This example is foreign example. Let me use some desi example which I always relate to. Okay, happened yesterday night only. Yesterday night only it happened. Okay, I take a nice plate, hot rice, tupa, like ghee on top of it. Nice ghee I will add. Other side I'll keep nice rasam. I'll keep one pickle is there. Just rasam with pickle extra. Okay, I'll keep one papad next to it. Entire mood is set. I'm watching a movie. I take a first bite into my mouth. As soon as the food goes into my mouth, my entire mouth burns up. Within a fraction of a second, the food is outside my mouth. Then I'm very sure this thing happens to all of you. You cannot wait for the entire food to go into mouth. But unfortunately, the rasam was too hot to handle. And the food is out of your mouth. And you don't even want to throw it out because it's so tasty. You want to gulp it down, but it is not possible because it is so hot. Right? It is so hot. That is basically your brain telling you, you stupid person, have some patience in life. If you give 2 minutes oof, if you do 2 minutes oof, the entire food is going to be cold and eat that time. But you don't wait. You take it in, your body responds and the food is out. What do you call that? We call that response to stimulus. We are taking the food in, the food is too hot. That is basically stimulus, the temperature, and you are reacting to it. We are reacting to, students. We are reacting it with the help of sensory organ. This is the mouth which I used yesterday night. Okay, so that is called as response to stimuli. This is a famous example. This is one famous example given here. Hot object, keep your hand is outside. Okay, I don't think anyone is stupid enough to do this. Okay, gas and all, who will keep? I can see the gas. I can see the gas is burning there. Why will I keep my hand there? Okay, if it is some patre, I can understand. If it's some utensil, I can understand. Right, utensil is hot. I would not want to keep my hand there. But who will keep a hand in the burner? Okay, so hot. So every single organism shows response stimuli. For example, this one, our famous plant also responds to stimuli. See, like how we look at a nice car is going, we'll see the car. Bike is going, we'll see the boys. Okay, right. Plants also, every single time the sun is up, they will keep looking at the sun and they will grow towards the sun. What do you call that, students? What do you call that? Right, like a plant which is growing towards the sun, I will call it photoperiodism, right? Photoperiodism, right? Or phototropism, right? That is response to stimuli. So every single organism does it. right? Not only us, right? And there are no exception. There are no exception here. So I will call this also as consciousness, also as defining. Okay, consciousness also as defining. As defining. And let me tell you, students, let me tell you, all of these features, right? These three mainly are found in all organisms. They are found in all the organisms. Okay. Now, apart from consciousness, students, there is something called as self-consciousness. How many of you know that? How many of you know that, students? I'll give you a simple example, all the boys, because I know it happens. As soon as you open your front camera, you're in a video call meeting, or you see one nice reflective surface, one glass anywhere. What do you do instinctively, students? Tell me. All the boys can tell me the answer. As soon as you see a reflective surface, without even knowing, you'll take your hand, you'll do this, right? You'll like, oh, nice hair, no? We'll fix the hair now. The hair is looking nice. Instinctively it happens. Now that is called as self-awareness. And let me tell you, students, self-awareness, right? It is only found in the stupid humans. Your animals do not have the self-awareness. Like a tiger will never ask, one stripe is missing. A zebra will be never like, oh my god, I'm not looking at a zebra crossing today. A giraffe will not be like, my neck is looking a little short today. A leopard will never be like, one spot is missing. No. Only we have it. Humans, called as self-awareness. Okay. So be careful of that. Self-awareness or self-consciousness. Now here one famous question is there. Is the person in coma also does he or she also have consciousness? The answer is yes. They may not have the consciousness like us, right? For example, if I go stand in front of a person who is in coma and start singing, start showing them nice biryani, they will not get up and start eating the biryani. Not possible. Okay. So if a person is in coma, they may not be responding to stimulus like us, but they are responding to the medicine which we give. They respond to the medicine. So I will call that cellular consciousness. A person in coma will show consciousness. There are no exception. There's no exception. Okay. And remember, students, the self-consciousness, right? Self-consciousness is a fundamental, because there are exceptions, right? Self-consciousness is going to be fundamental. Okay. So with that, students, your first part of the class is over. Actually. Now we go to the second part, which is actually even more easy. So with that, can we go to the next part of the class now? Can we go to the next part of the class today? So fast it gets getting over. Now, students, next part is called as nomenclature. Now, students, if I keep a cat here, right? If I keep a cat on the table, what will you call the cat as? For example, both of us do not know English, and I am going to call in my own mother tongue. Now, tell me, students, what do you call cat in your mother tongue now? Tell me in the chat quickly. Tell me in the chat, all of you. What do you call the cat in your mother tongue? For example, in my mother tongue, Kannada, I will call it Bekku. Bekkin Mari or Bekku, I will call it. Okay. All of you might have your own thing, which in own native language, some name. Okay. Now, students, the question arises: is there any solution to it? Because this person, let's say, is from Italy, like gatto, cat, catus, 𝐂𝐡𝐞𝐤 𝐿𝐻𝐿 Mahindra Bank. No. Kotaka. So everyone has their own name, right? Everyone has their own name. Pillai. Pilli sounds nice. Okay. Now, students, that becomes a problem. Now, the answer is, the question is there any answer to this? Is there any solution to this? Like, instead of having some random names for every single organism in our own mother tongue, can we find a solution to this? Is there any solution? Yes, there is. The solution is called as what, students? The solution is on the board. The solution is called as, right, it's a solution, right? Solution is called as binomial nomenclature. Okay, and this solution was given, not Augie. Augie is on cartoon. I go, cartoon you're telling. Okay. Now, binomial nomenclature solution. Binomial nomenclature was given by Carl Linnaeus, your great uncle. Okay, great uncle, binomial nomenclature. Okay. Now, what does binomial nomenclature tells you that? Okay. So, meaning of bi means two. So, every single name can be divided into two parts. Okay. First part is going to be genus. Second part is going to be species. Okay. Species. For example, Carl Linnaeus here. All of you, namaskara. Quickly namaskara here. Quick namaskara. All of you. Always respect should be given to the elders. Okay. Generic name or species name. It can be species name also. It can be species name, and all of this work was given in Systema Naturae and Species Plantarum. Those are the books. Okay. I'll give you a quick example. See this. Quick example. Okay. Mangifera indica. Mangifera indica, Linn. This is the scientific name. This is the scientific name of what, students? Indica car. Indica car. Tell me. What is the scientific name of? Indica. So indica means not indica car. Indica basically means Indian thing. Right? Not Indian, basically indigenous. Right? So Mangifera indica is the scientific name of mango. And this Mangifera here is the generic name or the genus. Indica is the specific name or the species name. Species. So Mangifera indica. Now what is Linn here, which I have not underlined? What is Linn? L-I-N-N. Linn. Not the big mom. Who is Linn? Linn is basically, it's called as author citation. We are giving respect to the author who gave this terminology. Right? Linn here refers to Linnaeus. Carl Linnaeus, the person who gave this name. So Linn refers to the Carl Linnaeus here. Okay, Linnaeus. Okay. Now, students, so a few rules here. We have to follow the rules. If you don't follow the rules, no marks given. No marks. Forget the marks. Okay. So let's see the simple thing here. Biological names are generally in Latin and written in italics. They are Latinicized or derived from Latin, irrespective of their origin. Right? For example, I find an organism. Let's take one Vignesh. Vignesh finds one new cat on the town. New cat he finds. Or new species he finds. He will call it Vignešius. Okay. But the problem is, he can't keep the cat name or the new organism name as Vignesh only, based on his name. He has to change his name, which is going to give it to the organism, into somewhat italic name. So you have to, it's a Latin name, Latin name. So he has to change the name of the new organism into something Latin. That is called as Latinization of the name. Okay. Now, the first word in the biological name represents the genus, while the second component denotes the specific epithet or species. Both the words in biological name, when handwritten, are separately underlined or printed in italics to indicate their Latin origin. They have to be in italics or underlined. The first word denoting the genus starts with the capital letter, right? And the specific epithet starts with a small letter. Okay, small letter. Example, Mangifera indica. Done. So finally, every one of them, right? Every one of them will call it Felis domesticus. That is the scientific name of cat. Okay, scientific name of, look the way it's sitting. Kinder Joy cat. It is Kinder Joy cat. Okay. Now, students, with that we come to something called as taxonomy or taxonomic categories. Now, say all of you have some idea right now about binomial nomenclature. In binomial nomenclature, we take one organism and we give it a name. Okay. Now, students, one small thing for you, all of you to understand. All of you understood about binomial nomenclature. Is there something called as trinomial nomenclature or monomial nomenclature? Mononomenclature or trinomial, quadrinomial? The answer is yes. There is something called as trinomial nomenclature. In fact, if you go to any of the famous zoos in the world, and whenever they keep a labeling of animals, generally animals, they use trinomial nomenclature, not binomial. They use trinomial nomenclature. If you're in Bangalore, if you go to Bannerghatta Zoo, that time you will see trinomial nomenclature there. Personal observation. Okay, so what is taxonomy and taxonomic category? Now students, I'll tell you very simple words. Very simple words. Okay. All of you close your eyes. Close your eyes. Okay. I will say an animal name and you should think of that animal in your head. Start. Can we start? Okay, close your eyes. Cat. Image came to your mind. Dog. Image came to your mind. Giraffe. Image came to your mind. Mango. Image came to your mind. Rice. Image came to your mind. Yes or no? Now all of this image which came to your mind is based on certain data, right? Now I will give you one more example. Okay. Skeleton flower. Did something come to your mind? Not really. If you do not know what is skeleton flower, wait for the short which is coming up. I have made a short for it. I have started a fact series, so that will come up. But when I told cat, when I told dog, when I told giraffe, when I told rice, mango, some image came to your head based on certain previous data, right? Based on certain previous data, you are able to tell cat, dog, rice, mango. Now students, you have 4 options with you. You have cat, dog, rice, mango. Now I will tell you, you are supposed to write plants separate, animals separate. Will you be able to write? Yes or no? Tell me in the chat. Yes or no in the chat. Tell me. Will you be able to tell me how many of them are plants? How many of them are animals? You can tell me. That is called as taxonomic categories. Based on certain data, you are able to tell something is animal and something is a plant. That is called as taxonomic categories, and that is a little complex eventually. Eventually it becomes complex. I will teach you that also today. Okay. Now for example, animals, mammals, dogs, all the good boys are here. Can you see? Animals, mammals, dogs, all of these are called as separate categories. They are called as separate categories. Yes or no? Tell me. They are called as separate categories. Now students, can I call all of these categories as also as rank, different ranks? There is a other name for this rank. The other name for this rank, it is also called as taxa. It is also called as taxa. So taxa is also called as rank, also called as category. This category, this category, this category. Like in army we have, right, lieutenant colonel, captain, havaldar, soldier, different categories, different ranks. Similarly, in the case of your animal hierarchy or organism hierarchy, like this, there are multiple categories, multiple ranks, and they are called as what? Taxa. Called taxa. So before we go to the entire taxa, let's understand the terminology, something called as taxonomy. What is taxonomy? Classification involves ordering of organisms into groups, branch of study that deals with principles and procedures of identification, classification and nomenclature of organism is called as taxonomy. Let me write for you. What exactly do we have under taxonomy? Under taxonomy, we have something called as first step is called as characterization Characterization, students. Characterization. Second thing is called as identification. Identification. Third one is called as classification. Classification. And the last one is called as nomenclature. All of these are different steps. All of these are different steps of taxonomy. For example, in characterization, under characterization, you basically observe. And you find certain morphological features. For example, when I say cat, 4 legs, furry tail. If you go near, it will bite you. Right? It will hiss at you. Or it'll scratch you and run away. Observations. That is called observation. Next thing is called as identification. In identification, we learn that what type of organism is this? What type of organism is it? Organism. After you identify it, after identification, next step is classification. You will keep it under kingdom, class, order, family, genus, and species. Based on that, it's called as classification. Now, after classifying it somewhere, right, you will give it a name, proper name you will give it. That is, from here, what is the genus? First one. Species from here you will take and you will give it a name. That is called as taxonomy. Very simple, no? Yes, it is. Taxonomy. Now, there is one more called as systemics. What is systemics? Nothing, very simple. Taxonomy is there, right? With taxonomy, just add certain evolutionary, right, relationship. Who is the father? Who is the great grandfather? Who is their father? If you add the evolution relationship plus taxonomy, that is called as systemics. Right? That is called as systemics. That's all. Systemics. Okay. Now here's a small different types of taxonomic categories: kingdom, phylum, class, order, family, genus, and species. Now, students, I want next 5 minutes. I want ultimate attention. Can you give me now? Next 5 minutes, the ultimate attention, where I'll teach you some very important concept from where the questions come, and probably the only confusing part about this chapter. If you learn this part, everything is going to be easy to you. Can we start? The next 5 minutes, that's all. Next 5 minutes, and after that, you're good to go. The chapter is almost over. At this point. Can we go? All of you? Can I get that undivided attention? Now for the next 5 minutes? Okay. Maximum effort now? Okay. Now understand. Tell me, students, I'll ask you simple questions. Tell me the answer. At kingdom level, first of all, this is the entire classification. You need to learn this order. You have to learn this order. Right? Keep pots clean or family gets sick. All of you know that, right? Keep, not mine, pot clean or family gets sick. Keep pots clean or family gets sick. That is kingdom, phylum, or division in the phylum in the case of animals, division in the case of plants, class, order, family, genus, species. Learn this much, all of you should know. This. Next step you need to know is, in the case of kingdom or in the case of species, where do you think we have more number of organisms? More number of organisms can be found at kingdom level or species level. Obviously kingdom, plant kingdom, animal kingdom, fungi, Monera, Protista, right? So more number of organisms at this point here. More number of organisms. Here we will have as we go down and down, as we go down and down, number of organisms is going to decrease. Number of organisms is going to decrease. Okay. Now, students, if I ask you a simple question, which organisms have unicellular organisms but prokaryote? Which kingdom comes to your mind? Which kingdom comes to your mind when I say unicellular prokaryotes? Kingdom Monera. Unicellular eukaryotes, Protista. Saprophytic mode of nutrition, fungi. Photosynthetic mode of nutrition, plants. Heterotrophic mode of nutrition, animals. All of you agree? So these are called as general characteristics. General characteristics. Unicellular prokaryotes, unicellular eukaryotes, photosynthetic in nature, saprophytic mode of nutrition. These are called as general characteristics. And these general characteristics are found where? More at the kingdom level. More at the kingdom level. General characters. But as we go down and down and down, the general characters are going to decrease. The general characters are going to decrease here. It's going to decrease. Okay. Now, probably the important one is called a similarity. Now, students, I'll give you a simple example. Tell me. Let's take sponges, sea anemone, a fish, right? A fish, a bat, a human, a cat, a dog, donkey, a lion, a cheetah, right? A cheetah, right? Let's take a mollusk. Do we have any similarity among all these organisms? Do we find high similarity or low similarity? Tell me. Between sponges and humans, sea anemone, fish, bat, mammals, do you find more similarity or less similarity there in all these organisms? Very less similarity. I do not look anything like sea anemone. I do not look anything like a sea fish, a shellfish. I do not look like anything like that. So at kingdom level, animal kingdom, there are less similarities. But as we go down to a species level, humans, species level, genus, different types of closely related species of humans. Let's take different types of dogs, right? Different type of cats. Do they have more similarities among them? Yes, they do. So as we go down and down and down, what happens? There is a increase in similarities. So as we go to the species level, it will have the highest similarities. As we go down and down and down. Similarities are going to be increasing as we go down the entire hierarchy. For all the way from the kingdom to the species, similarities are going to increase as we go down. It is going to increase as we go down. Okay. And remember, students, species is technically basic unit of classification. Basic unit of classification is going to be species. It is going to be species. And let me know, students, if you want these notes, do you want these notes? I know I have not submitted the previous notes also. So today on my Telegram channel, Baswaraj Sir Biology, all of you are aware, right? In my Telegram channel, you will be able to get, I'll write off. On this Telegram channel, I will be uploading all the notes. I'll be uploading all the notes here. Remember that. Okay. So I believe all of you understood this. If you understood this, chapter is over actually. Chapter is over. Okay. So as we go down and down and down, the similarities are increasing. Can you see, students? Can all of you see from kingdom level, a lion, giraffe, this orangutan, elephant, panda, right? Similarities is not there. But as we go down and down to to the species level, do you find similarities are increasing? Are they increasing, students? Similarity is increasing here. It is increasing here as we go down and down. Simple as that. Okay. Now, students, there's something called as taxonomical categories. So there are something very common among species, order, phylum, or division. Now we'll understand that. Small part, 15 minutes, chapter is over. 15 minutes, 10 minutes, chapter is over. I bet. You don't require more time than this in this chapter. You don't have to invest more time than this. Whatever I've taught you, you learn this much, you're good to go. You're good to go. The only thing which is missing right now, which I haven't told you, is a definition of ICBN or ICZN. I'll tell that in the end. Okay. Now, what is species? A group of individual organisms with fundamental similarities. Lowest taxonomic category we saw here. Species. Done. Now, species. Okay. Tiger, lion. That is, both are different type of species. They have good similarities. Now, what is genus? One more example is here. Solanum tuberosum, Solanum melongeni, that is Protozoan brinjal species. Example species. Now, what is genus? Understand what is genus now. Understand what is genus. Where's the brightness? Okay. Understand what is a genus. What is genus? Okay. Now, I'll give you a simple diagram to understand genus. Okay. Let's take, here we have genus 1 and here I have genus 2. Genus 2. Okay. Each genus has multiple species. Each genus has multiple species. 1, 2, 3, 4. All of these species will make up one genus. All of these species will make one more genus. And all the species here will have certain common characteristics. That is why they are grouped together. They are grouped together. See, group of related species having more common characters in comparison to species of other genera. Okay, they are grouped together. Okay, they are grouped together. This is one genus, genus 1 and genus 2. Okay, genus 2. And students, that is basically genus. Now, what is family? Okay, what is family? Understand. Multiple genus which have common characteristics. Genus of a cat and dog, right? When they join together, they make up the family. Okay, group of related genera are more similar to each other than the other family. They make up the family. Multiple genus together is called as the family. Okay, family. Now, students, again, multiple families make up the order. They make up the order. One example is given here, students. Understand. Panthera tigris, Panthera leo. They are species, but they are under same genus. Panthera, same genus. Panthera. But the genus of big cat, genus of small cat. I made it right here, right? Genus 1, genus 2. Genus 1 is, let's say, is big cat. This one is small cat. Small cat. Okay, small cat. Are they belonging to same family? Same family? Yes. They belong to same family called as Felidae. They belong to same family called as Felidae. So, related genus coming together to form the family. Family Solanaceae, which has genus Solanum, genus Datura, genus Petuna. Multiple genus coming together to make one entire family. Okay. Now, families come together to make the order. For example, cat family and dog family. There are two families. So, when two families join together, what do we get, students? We get the order. Okay. Polynomials is the order. Solanaceae and Convolvaceae, both are the different families. They join together to make up the order. Okay. Order. Now then we have division and then kingdom. What is division? Multiple different orders join together to make up the class, right? To make up the class, later orders. Multiple class join together to make up the phylum. Then we have kingdom, that is five kingdom classification. Five kingdom classification. For example, class Mammalia, all of you are aware, right? Primata and order Carnivora, both are belonging to the mammals itself, that is our ancestors and non-vegetarian cousins. Both are different orders joined together to make a class. Okay. This is a taxonomic hierarchy we just now learned. Species, genus, family, order, class, phylum or division. Phylum in the case of animals, division in the case of plants, and make up the kingdom. They make up the kingdom. Example given for human here. Example given here for the human example given here. Okay. Now this is a table which you have to remember at any cost. Multiple questions have come year after year. Multiple questions have come year after year from this table right here. So I'll give you exactly 30 seconds to see this table and then ask you questions. Your time starts now. Your time starts now. I'll give you exactly 30 seconds to see the table. 30 seconds. Done? Done, students? While you read it, let me write some two definitions which you have to remember. Okay, or full forms, not definitions, full form. Okay. First one is called as international, right? International code for botanical nomenclature. Second one is called as international code of zoological nomenclature, that is ICBN, International Botanical Nomenclature, ICZN. Okay, these are two different definitions, two different full forms which you need to remember from this chapter. International Code for Botanical Nomenclature will give all the plant names. International Code for Zoological Nomenclature will give all the animal names. Will all give the animal names. Okay, so this you have to remember at any cost, students. Let's go over it. Man, Homo sapiens, all of you know. Homo is the genus, species, sapiens is the species. Okay, species. Now, family is Homidae, Primata, Mammalia, Chordata. This all of you should be knowing. Students, this is the one which is mostly asked, housefly. That is Musca domestica, Musca, Muscidae, Diptera, Insecta, Arthropoda. Mango, all of you should be knowing. Mangifera indica, Anacardiaceae, Sapendales, Dicotyledone, and Angiosperm. Triticum aestivum is the wheat. Triticum is the genus. Aestivum is the species. Poaceae is the family. Poales is the order. Monocot and Angiosperms. You have to remember this at any cost. You have to remember this. Now here is a small question to all of you. Let's see who can solve this question. Okay. Students, quickly answer in the chat right now, and I want you to keep a track of your marks. How many marks you will get at the end of it. Okay. Quickly, all of you, quickly tell me the answer in the chat. Match the column of 1 and 2 for the housefly classification and select the correct option using the codes given below. Family. Muscidae is the family, yes or no? Let's see who does first, me or you. Okay. Family is gonna be Muscidae. Order is gonna be Diptera. Order is gonna be Diptera. Okay. Order is going to be Diptera. Class is going to be Insecta. Phylum is going to be Arthropoda. So A is going to be 3, B is going to be 1. Option 1. Option 1. Next question. Housefly belongs to dash family. Housefly belongs to dash family. What is the family name of housefly? What is the family name of horsefly? Done? Done. X and Y are the components of binomial nomenclature. This naming system was proposed by Z. X is the starting one, students. X is the starting one, right? I got my answer already. Generic name, specific name, and species epithet. Generic name, species specific epithet, and car lineus. In taxonomical categories, which hierarchical arrangement is ascending order in correct sequence in the case? Ascending means from the larger to smaller you have to go. Top to bottom. Tell me. Kingdom, phylum, class, order, family, genus, species. Students, very easy. Very easy. Which of the following belongs to family Muscidae? Again, housefly. Same question has been repeated again and again. Next question. Which of the following is against the rules of ICBN, International Code for Botanical Nomenclature? Against it. Against it. Handwritten scientific name should be underlined. True. Every species should have generic name and specific epithet. Yes, true. Scientific names are in Latin and should be italicized. Yes. Generic and specific name should be written starting with small letter. No. That is, generic name has to be capital. Should be cap. Okay. Nomenclature is governed by certain universal rules. Which of the following is contrary to the rules of nomenclature? The first word in biological name represents a genus and a second specific epithet. True. Names are derived from Latin or written in italics. True. When written by hand, their names are underlined. True. Biological names can be written in any language. Not true. They are supposed to be only in Latin. Only in Latin. Okay. Next question. ICBN stands for International Code for Botanical Nomenclature. Easy question. I just don't want it. Which of the following taxonomic categories is less general? Where do you find less general characters as compared to genus? Students, tell me. As we go down, general characteristics are lesser. Right? I told you, general characteristics are always more at the kingdom level. As we go down and down, general characteristics will decrease. But similarities will increase. General characteristics will decrease. So at the below genus is what? Below genus is species. So at the species, very less general characteristics. Very less general characteristics, students. At species level, very less general characteristics. All of you remember this table? See here. Very less general characters at the species level as we go down and down. Okay. Last question, students. Last question for today. Last question for today. And I want this is the last homework question also from my side. Homework question from my side. Okay. This is the homework question. I want you to tell me the homework question. I want you to tell me the homework question in the comment section after the class, and also tell me this chapter is done and dusted. That's all. Chapter is over. Now students, do you want a 5 minutes break before Kovika ma'am class? All of you, do you require a 5 minutes break before Kovika ma'am class? Let's take right now. Time is the time is. Students, you realize the chapter got over so fast before you could realize it. The chapter got over before you could realize it. Right, chapter got over. Now the time is, let's say 6:20, 6:25 ma'am will start. Class resume at 6:25. Class resume at 6:25. Now students, listen to me very carefully. Right? Listen to me very carefully. If you want these notes, you can get these notes right after the class, even in the description. if you go to the description of this video, you will be able to find all the notes today itself. How does that sound? Right? If you want to download these notes, it is available in the description. So download today only and revise one more time, and nothing can stop you from scoring out of 40 in biology. Right? With teachers like us, we'll make sure you don't even lose a single mark. So download the PDF today only. Okay, deal. All of you, deal. Yes or no? All of you should download the PDF of this chapter as well as biological classification today itself. Both of them today itself. Let us know in the comment section how was it to be part of Bio Club. Right? I had so much of fun with Bio Club. It was so fun with Bio Club. Right? And I believe all of you love biology more. Right? I hope that. Okay. So I will give a 5 minutes break, and let's start the break, and I will see you in the next video. Until then, take care, students. Bye bye. And that is, I am out of here. Bye bye. Let's wait for Gopika ma'am. Don't run anywhere. Wait for Gopika ma'am.
Hello students, welcome to Vedantu NEET English. I am Ms. 𝐂𝐡𝐞𝐤, your own biology master teacher. Welcome all of you to Biology 360 Club, and it might be the last class of Biology 360 Club, but there's nothing to be sad about because it's not the end of anything. It's the start of another thing. That is, we are going to start some backlog series for you and also some practice sessions for you because we have 3 months left for NEET. Right, not exactly 3 months, 3 months and few days left for NEET. So we will be making sure that you get enough practice sessions. Also, go back and finish all your backlogs, right? All of you are ready? Let me just inform in the Telegram as well, okay, because many of them has gone for break. Yes. Done. Yes. All GMAs are here. Okay, shall we start? Yes. So students, biological classification, as all of you know, we have done this chapter before also, and it is very easy to understand that point of time, but later when you want to revise it, the chapter becomes a little complicated for all of you, right? You're not able to remember, you're not able to revise all this. So today we are going to do like a revision, as well as I will make you understand for those students who have joined first time also. Okay, so students, understand that the session PDF, okay, after the class my notes will be available in the description box. So after the class, once the notes are updated, I will share it on my Telegram, that is Biology by 𝐂𝐶𝐞𝐤, and on Vedantu NEET English Telegram once the notes have been updated. And also in the description of the video you will get practice questions of living world and biological classification. Okay, right? Yes or no? What happened? Yeah, did ad play or something? Or am I not live? Oh yes. Okay, something happened. Suddenly students disappeared. Yes, so you will get the PDF, so you do not have to worry about. Now students, let's go to the introduction of biological classification. Why do we need classification? What is the necessity for classification? That is the first thing. See, to study one or two organism provides the idea of the entire group. If classification did not exist, okay, if the class, yes, generally this will be the last session of Biology 360 Club, not the last session on Vedantu NEET English, okay. Biology 360 Club, we have done all the major chunk of the units. If you notice, all the high weightage units we have completed. Now human physiology, plant physiology, me and Baswa sir cannot do it together, so we will be taking it as separate entities. Okay, do not worry, entire portions, as I promised you, Biology 360 is still the goal, so we will be doing it separately. You will not see Baswa sir and me together on one live. Okay, yes. Perfect. Ecology unit also Baswa sir has recently done it. You can go and check it out. Benny, you can go check out the ecology unit as well. Now to study, see, what's the use of classification? If I learn about one or two organisms, I will be able to understand how the entire group is. Now when I'm teaching you about Monera, even though I'm telling you the sole members are bacteria, you're not learning about all the bacteria, right? We are just studying the general characteristics. Same thing goes with fungi, same thing goes with plantae, same thing goes with animals, right? We do not study all the organisms. We study one phylum and we are like, okay, all of them have similar characteristics, one or two exceptions will be there. So that is the first use of classification. Helps in identification of an organism, helps to identify the relationship between different groups of an organism. You will be able to know which are in close similarity, you will be able to know which are away from each other. You will also be able to know, oh, this organism and this organism looks the same, but internally they are very, very different. All this is possible again because of classification. Helps in knowing the evolutionary relationship between organisms. Evolutionary history means what? About their ancestors, phylogenetic relationship. Okay, clear to all of you? Yes. Now students, classification, when we talk about, please understand that classification, just one day, you know, R.H. Whittaker came and he's like, okay, let's do 5 kingdom classification. It did not come like that. It started on a very non-scientific pattern, and then slowly it went to scientific pattern. Okay, all of you call your friends here. I'm very disappointed with the crowd. This is not what you people promised me in the starting of Biology 360 Club. And I know some of you are very regular, but then some of you are very like, you know, oh ma'am, Saturday. Students, for you people know Saturday, Sunday, right? For me at this age, if there is no Saturday, even for you at this age, there shouldn't be a Saturday, because this is not the time for you to sleep. sit and watch family movie time, because for your parents, yes, Saturday and Sunday might be off, or Saturday half day they might come, so for them it's enjoyment. For us it is not, right? We are preparing for a competitive exam. Thank you so much, Madhumita. Yes, so earliest classification started on a non-scientific note, and it was given by who? It was given by, so first classification, it did not come by any people, but it was generally based on three things: food, shelter, and clothing. So first ever classification started because of these three aspects, that okay, you are giving me some food, so you are going to fall under food. You are giving me some jute or some cotton, you are going to be put under clothing. You are giving me logs or wood, you are going to be put under shelter. But this kind of classification, is not what we are expecting, right? This is not the classification that we are expecting. That is, just by looking at something, how can I just classify them? I cannot classify them. So just by looking that, okay, you're giving me food, I'll put you into this category. You're giving me shelter, I'll put you into this category. This was not possible. So this classification was not widely accepted at all. And this was done merely for people's greed and need, students. It was not done for any other thing. It was merely for people's greed and need. Okay? Greed and need. That is the main reason. Okay, and this was not accepted. This was not accepted. Then came the first ever scientific classification. First scientific classification by who? Aristotle, who is father of biology and zoology. Okay, father of biology and zoology. Clear or not? So students, the first ever classification came from Aristotle, who is father of biology or zoology. And what was his classification based on? His classification had something more to it, okay? His classification had something more to it. That is, he told that I am going to do classification based on, in plants, I am going to do based on their habitat, or just how they are at their sizes and how they are categorized, okay? That means plants I am going to divide based on three categories. That is going to be herbs, shrubs, and trees. That means mere morphology. Just by looking at the morphology, I'm looking at you people and I'm telling, oh, I am here, you are like this, you are like this. That is not classification, right? That is me telling by myself that yeah, I can classify you people like this, right? So based on mere morphology, he classified plants into herbs, shrubs, and trees. Okay, herbs, shrubs, and trees. Now what about animals, ma'am? He classified animals on the presence and absence of. Okay, he classified animals on the presence and absence of RBC, that is your red blood cells. He classified into what? Enema, and enema. Enema and enema. Clear or not? Yes, very good. Enema and enema. Enema, A, absent. Okay, absent. RBC was absent. Enema, present. So based on absence and presence, okay, based on absence and presence, he categorized the animals into enema and enema. A for absent, A for enema. Remember like that, okay? Enema and enema. So this was the first ever classification, student. The first ever scientific classification was given by Aristotle, and he is known as the father of biology or zoology. What was this classification? On mere morphology. He classified plants telling that, oh, you are a shrub, you're a herb, you're a tree. That's it, mere morphology. Then he classified animals little more in depth by the presence and absence of RBC, that is your red blood cells. He called it enema for those who did not have RBC, and those who had RBC, he called it enema. Clear? No doubts in this? Okay, perfect. Perfect. Now students, this first, I would say, now lot of students, again this I have told you previously also, many students when they speak about two kingdom classification, very very carelessly some of you write Aristotle. Please understand, Aristotle was in the BC time. That means he came up with this classification very, very, very before. Okay, before you can even put it into your dates, no, like that. BC time he came. But the first ever kingdom classification, two kingdom classification, was given by Carl Linnaeus. Okay, so whenever the question comes, who gave two kingdom classification, your answer should be Carl Linnaeus, not Aristotle. Fine. Thumbs up in the chat if you understood that. Yes, you people should put fire emojis in the chat box. Then only it is going to go forward. More students are going to join. Okay. Yes. Come on. Clear? Very good. Carl Linnaeus was the person who gave two kingdom classification. Ma'am, on what basis did he do two kingdom classification? Students, he did two kingdom classification on the presence, okay, on the presence and absence of cell wall. On the presence and absence of cell wall. That means a more scientific way, right? Dealing with it more scientifically. That means you have cell wall. Okay, you are here. If you don't have cell wall, I will put you here. So that kind of thing was done by who? Carl Linnaeus, right? See, when your fire emoji comes early, my energy also comes. Okay, so keep, put energy in the chat box, because that's the only way we feel also, okay, from this way, that students are understanding, they are enjoying the class. Okay, yes. So students, presence and absence of cell wall, what did he do? He put so many things into plant, okay, without even bothering if they were autotrophs, heterotrophs, unicellular, multicellular, eukaryotes, prokaryotes. Carl Linnaeus is like, this is not my cup of tea. I will only see the presence and absence of cell wall. Because of it, what happens, friends? Lot of mix happen, okay? Lot of mix happen. Clear? So here you can see algae, fungi, bacteria, bryophyte, pteridophytes, spermatophytes, or your, okay, coelant, again, animalia they put, okay, lot of this is there, platyhelminthes, chordates, hemichordata. With this we can add amoeba, okay, paramecium. All these were added into animals. But now where are you studying all this? You are studying under Protista, right? Yes. Okay. So students, there's nothing to be emotional. Okay. This is just a course. Okay, it's just a course of time. So Biology 360 Club will be there, but I'm happy that I want you people to glorify the name. I want you people to tell the name out loud, so that, you know, we feel like, okay, we should come back with it. Okay, now if you see the crowd and everything, it feels very sad, no, that, oh my god, starting they promised so many things, now they are not, you know, staying back on words with that. So understand, but we are not going to stop anything in the middle, or we are not leaving it incomplete or anything. We are going to continue, just that me and Baswa sir together cannot do plant physiology and human physiology, because it does not make sense, no? Photosynthesis, after that I'll be taking breathing and exchange. So that is why we are going to separate it, okay? That's it. But we are going to be back here. We are going to take classes regularly. Okay, so presence and absence of cell wall. Carl Linnaeus came up with two kingdom classification, and you can see a lot of mixtures are there, because here you can see fungi is there. Fungi cell wall is made up of chitin. Okay, so everything was a confusion. But there was a person who was looking at Carl Linnaeus and telling that you are doing wrong. So there are some drawbacks of Carl Linnaeus that you need to study. What are the drawbacks? Looking at that only you understood that he did not differentiate so many things. What and all he did not differentiate? All of you tell me in the chat box. Most of you are studying this with me for the third time, so you should be able to answer. He did not differentiate. Yes, in the chats. Yes, tell me in the chats. Very good, very good. Who is that? Kubera, very good. Okay, he did not differentiate prokaryotes and eukaryotes. Okay, that means the ones which does not have a nuclear membrane, right? A well-defined nuclear membrane. The ones who does not have a well-defined, alright, NM, nuclear membrane, and the other one who has a well-defined nuclear membrane, right? He did not differentiate that. Second thing, what did he do, students? He did not differentiate unicellular and multicellular. He did not differentiate unicellular and multicellular. Can we agree to it? No, ma'am, we cannot agree. How can he not differentiate that? Next thing, very good. He did not differentiate photosynthetic and non-photosynthetic. Okay. All these notes will be available. Don't worry. And if you want to join my Telegram, you're always free to join my Telegram. That is Biology by Gopika. Okay. Yes, photosynthetic. So he did not differentiate prokaryotes with eukaryotes, and he did not differentiate unicellular, multicellular, photosynthetic and non-photosynthetic. The biggest example we can see is he has mixed, right? He has mixed, let's say, higher plants. Okay, like your pteridophytes. Okay. He has kept pteridophytes and fungi together. Is this okay, students? He has kept these two people together. Is it okay? Of course not, ma'am. One is capable of making its own food. That is, one can do autotrophic behavior, right? One is autotrophic, another one is completely heterotrophic. If you see the cell wall also, they are completely different, right? So he mixed everything together. He mixed everything together. So mode of nutrition was never even into his thought process. Okay, this was the problem of your Carl Linnaeus two kingdom classification. Then who was looking at him? Ernest Haeckel was looking at him. And who is Ernest Haeckel? We have studied it in evolution, biogenetic law. Even though the biogenetic law was completely rejected by Vaughan, but then Ernest Haeckel still gave the rule, telling biogenetic law he gave, and also he gave up the three kingdom classification, right? What is three kingdom classification? He was like, Carl Linnaeus, what are you doing? You cannot do all this, kind of kitchery. So I will give one more kingdom, okay? So which was the already existing kingdom, students? Two kingdoms was already existing from Carl Linnaeus' time, that is kingdom Plantae. Okay, I'm just writing it like this. Okay, Animalia. Okay, Animalia. He added, Ernest Haeckel added which kingdom? All of you tell me in the chat box. Quickly tell me in that chat box. Very good, Meghana. He added Protista. He added Protista. Telling that, okay, let me add Protista. Now, what was he intending by adding Protista? He was like, let's do one thing. Let's put all the unicellular organisms here. Okay? All the unicellular organisms, right? That was his plan. Yes or no? Very, very good. Sneha, Karti, Manu, yes, Roshni, very good. What did he tell? Let's do one thing. Let's just put all the unicellular together so that Carl Linnaeus' one mistake can be, rectified. Right, rectified that, okay, unicellular, multicellular together. So let's put all the unicellular. But still, it did not solve everything, students. Then came four kingdom classification. Again, four kingdom classification was given by Copeland. Okay, four kingdom classification was given by Copeland. So kingdom, again, Plantae existed. Okay, Animalia existed. And as of now, Protista also existed because Ernest Haeckel had told about it. Now, a new kingdom that came into existence was Monera. Monera. Now, Monera, what was the plan? He was like, let's do one thing. Let's put all the prokaryotes here. Okay? Prokaryotic organisms here. So, now two mistakes we were able to rectify. What was that? All the unicellular we separated, now prokaryotes also we separated, okay? Again, there was upside downs. Always remember these changes are going to be your permanent thing in classifications, because every time some new organism comes and they notice that, okay, this is going here, this is going here, they are going to change few things. And also identification of some organisms, going back to the phylogenetics, they are going to be able to change few things, okay? But as of now, this was what Copeland told us. We will introduce Monera, kingdom Monera, and in that Monera, let's put all the prokaryotic organisms. Then comes the most relevant, widely accepted, okay, most relevant, widely accepted classification by R.H. Whittaker. Now, students, R.H. Whittaker went into what? R.H. Whittaker was like, I will go into the depth of understanding what is there and what is not there. That is, I am going to see the cell type, I am going to see the cell wall, I am going to see the nuclear membrane, body organization, mode of nutrition, and also, very importantly, he also went back to the phylogenetic relationship. He also went to phylogenetic history. That means what, students? He went back to the ancestors. That is what? Evolution. He learned the evolution. And who was this? This was done by R.H. Whittaker. Okay. This was given by R.H. Whittaker. And the question can come, which is the most widely accepted classification? Your answer should be R.H. Whittaker's 5 kingdom classification. Even though there is 6 kingdom classification, still the most widely accepted one is R.H. Whittaker's 5 kingdom classification. Okay. Now students, let's see what exactly made this classification so special, or why did everyone actually come into accepting this. Okay. Look here. First, he categorized them into cell type. What did he do? Monera, Protista, Fungi, Plantae, Animalia was there. In that, we told already that in Monera, we will put all the prokaryotes. That means the ones which does not have, okay, the one which does not have nuclear membrane, okay, and they just have like nucleoid kind of structure. Okay, nucleoid kind of structure where there is no well-defined boundary for the genetic material to be there. Okay, clear? Very good. Okay, yes. So he told this. Then the next thing was non-cellulosic polysaccharide plus amino acid. Okay, so the cell wall in Monera was non-cellulosic. That means it was not made up of cellulose. But what was it made up of? Polysaccharide and amino acid, or you can write polysaccharide amino sugars also you can write. You can remove that polysaccharide word and you can just write amino sugars, or else in other you can also write peptidoglycan. Peptidoglycan. Okay, peptidoglycan. This is also the name you can write. Okay, you can write any of this. Peptidoglycan you can write, or you can also write non-cellulosic. Also, you can write amino sugars. All this is accepted. Okay, you can also write amino sugars. This is what it was made up of. It was non-cellulosic. That was clear to all of us. Okay. Now, students, if you come further, obviously nuclear membrane is absent. That is why it is a prokaryote, right? So here the nuclear membrane is absent. Now, body organization is cellular level, because these people are very primitive. Very primitive. Now, look here. Mode of nutrition, very, very important in case of Monera. Why, ma'am, so important? Because these people are capable of doing autotrophic. Under autotrophic, they can do 2 categories: chemosynthetic and photosynthetic. Okay, what can they do? They can do chemosynthesis and photosynthesis. Okay, clear? Now, heterotroph, saprophytic or parasitic. That means they feed on dead and decayed, or they will depend on another organism and they'll take all the nutrients from that organisms and be a harm to the host. Okay, be a harm to the host. Clear? Yes, did you all like the video? If you like only, YouTube will know that you are interested in NEET, not in any other gossips. Okay? Yes, moving forward, students. Protista, eukaryotes, present in some. That means in some cell wall is there, in some cell wall is not there. And again, Protista, when you are studying, the major questions can come from the cell wall itself, because one has pellicle, one has cellulosic plate, one has overlapping soap-like structure. So all these questions only will come for you in Protista. Now, students, nuclear membrane is present. That is why it is eukaryotes. Here also they are cellular. Little primitive itself, these people are. Okay, these people are little primitive. Here you can see they are autotrophic as well as heterotrophic. Again, photosynthetic in nature, but the only one who is capable of doing chemosynthesis in the entire kingdom is Monera. Okay, that means they use chemicals to yield energy. Okay, they can do nitrogen fixation. All this is capable of who? This people. But again, legumes also do nitrogen fixation. But here main thing is they take up the chemicals to release energy or yield energy. That is very important. Now, moving forward, fungi you have Here, eukaryotes obviously present with chitin. That means the cell wall is made up of chitin. Okay, multicellular loose tissues. Here you can see slight development is happening. Yeast is an exception, but otherwise you will see multicellular loose tissues are there. Heterotrophs, obviously they are not going to be autotrophs. We are moving towards that, no, saprophytic, or they will be parasitic. Very good, very good. Okay. Present obviously in plants, cellulose is present, tissue and organ level. These people are purely going to be autotrophs. Again, some examples like insectivorous, that exceptions are there. Again, here it is absent, tissue, organ system. Here it is holozoic, that means we do digestion, right? We eat the food, chew the food, then we digest it, right? So that holozoic is there and saprophytic is there. Any doubts in this? Tell me in the chat box. Yes, tell me in the chat box. This box is only thing that you have to study for R.H. Whittaker. So you can all thank R.H. Whittaker with all your heart and soul, okay? All your heart and soul, telling that thank you for giving us just one box to study, because everyone else gave us one paragraph to study. Yes, the last 6 kingdom classification, which was given by Carl Woese. Okay, now ma'am, why is this kingdom not accepted? Because it did not have that much difference with your 5 kingdom classification. If I show you, you will understand. Students, here in 6 kingdom classification, the first word that you will come across is 3 domains. Tell me, is domain bigger than kingdom? Just now you studied living world, no? Keep the pond clean or the family gets sick. So in that you know that before kingdom, that means domain comes above, right? Domain comes above. So he made 3 domains. What was the 3 domains? He made domain 1 as RK. Okay, RK. Next was U. First I write bacteria. Okay, RK. Then he got bacteria. You can also write U-bacteria, not a problem. And he wrote eukarya. Okay, these were the 3 domains he came up with. Ma'am, but why did he do the separation? What was the need to do the separation, students? The need to do the separation was based on a 16 sRNA. Okay, he learnt about this and he noticed that, okay, Archaebacteria and U-bacteria cannot be put under one thing. So I will make it separate. Okay, I will make it separate. Clear? Okay. U-bacteria, eukarya. Very good. Is that clear? No doubts in that, right? So I'm telling you why he made that. Okay, why? What was the necessity to separate them? Was because he was studying on 16 sRNA, that means the ribosomal RNA. He also studied about their cell membrane. Okay, he also studied how their reaction is with some antibiotics. All this led to the conclusion that Archaea and U-bacteria cannot be put together. He understood that Archaebacteria and U-bacteria cannot be put together. Understood? Again, he did the lipid structure, 16 sRNA, many things. Okay, small things he learned. So I'm just to make you understand, I'm writing this. It's not necessary for you to remember. Okay, but just understand that why did he come to that conclusion is because of that. Okay, yes. Now further, students, under Archaea, what did he add? He added Archaebacteria. He put Archaebacteria. And under bacteria, what did he put? U-bacteria. Okay, U-bacteria. Under eukarya, he went further to put Protista, Fungi, Plantae, and Animalia. Clear or not? Animalia. This was the plan. So if you look technically, right? If you look technically, then okay, if you look technically, there is no much difference, right, with 5 kingdom classification. Just that one domain has come where he separated Archaea and U-bacteria, but otherwise 5 kingdom classification would be the widely accepted classification. Clear or not? Okay, yes, perfect. Thumbs up in the chat, this is clear. Okay, perfect. So 3 domains is clear. That is given by 𝐂𝐤. And why did he separate archaea and bacteria? Because of he was working on a ribosomal RNA, 16S ribosomal RNA. He also saw their cell membrane. He saw their lipid structure. He saw how they are reacting to antibiotics. He noticed that, okay, these 2 people have to be separated. Okay. Yes. Perfect. Perfect. Okay. Students, you people will give answer to 𝐂𝐤 or should I give the reply to the 𝐂𝐤? Yes. If you can give the reply, it would be good. Yes. Perfect. Okay. Rathis has given the reply. Now, moving forward, this is a box given to you. You can just revise through this box. Okay, you can revise through this box. As you can see here, students, kingdom Protista is added, all the unicellular organisms. Here, kingdom Monera was added with all the prokaryotic organisms. And then your fungi was added by R.H. Whittaker. That was the biggest change, right? If you look at the classification here, till Copeland, we were like, okay, Protista, we told Monera, we told. But what is the main thing that R.H. Whittaker came up with? Okay, R.H. Whittaker came up with. That is here, fungi. Okay, fungi. Clear? Fungi was the big thing he came up with because he was like, you people stop throwing fungi from here to there and there to here. So let's put fungi into a complete kingdom itself. So that problem is sorted because they are very special. They are so different. We are not able to put them anywhere. Okay, now students, issues solved by this 5 kingdom classification. As I told you, see, as a botanist, as a zoologist, okay, and people who study about different kingdoms, no, they will have their opinion that, okay, I want to study amoeba and paramecium somewhere else. I want to study Chlamydomonas cholera, okay, Spirogyra somewhere else. So this kind of conversations keep happening, right? So what was the issues mainly solved by 5 kingdom classification? Tell me in the chat box. Okay, 5 kingdom classification. Okay, yes, that is, some people, before what happened, amoeba and paramecium was placed under Animalia, right? It was placed under Animalia. And here, your Chlamydomonas okay, right, Spirogyra first. This were, many people love to learn it under Plantae. Okay. Now, all this was a problem, right? All this was a problem. Correct or not? Yes. So, this problem to sort, who was able to help? R.H. Whittaker was there to help. He told, let's put all of these people under Protista. Okay, all of these people under Protista. But again, many people did not agree to this. Okay, and again, there were some people who wanted to study it under Plantae, who wanted to study it under Animalia. But then R.H. Whittaker was like, no, let's not complicate it. Let's study all of this under Protista because they are going to fall under, okay, they are going to fall under Protista category, because on the simple basis of telling that, okay, you people are eukaryotes, let's learn all of these people together. Okay, so some of the issues that they were sorted were, I will show you in further. I'll show you further. Look here. Okay, earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm, angiosperm under plants. Okay, the character that unified this whole kingdom was all this organism included had a cell wall. So as I told you, Carl Linnaeus classification was merely based on absence and presence of cell wall. So he put all these people into one place, and those who did not have a cell wall, he put that side. Okay, but can you accept it like that? Of course not. So this was the first problem that was solved by R.H. Whittaker. It also grouped together unicellular organisms and multicellular ones together, like your Chlamydomonas and Spirogyra were placed together under algae. Even though you study it there, still people are like, no, how can these people come here? Let's put them there. So that kind of confusions were solved by R.H. Whittaker. Paramecium and amoeba, which were earlier placed in animal kingdom, which lack cell wall, it has been put together in organisms, and right now it is in Protista. Clear? Yes, in Protista. Which chapter today? What happened, Sony? Okay, now we will start with Monera. Ready, all of you? Ready, steady, go. Tell me in the chat box, all of you are ready? Okay. So students, Monera, first ever point that you have to study is that the sole members, okay, the sole members of Monera is bacteria. Is who? Bacteria. Sole members are bacteria. These bacteria are cosmopolitan in nature. What are they? Cosmopolitan. That means they're everywhere. You touch anywhere, they are there. Cosmopolitan. That means they are everywhere. They are good, some are bad, right? Some of them are inside our body, some of them are on my hands, on this pen, around this area, everywhere they are. Right now you will be like, ma'am, I just cleaned my room. But right now if I take a culture plate and come to your home and take a, like in the air, if I just do this with the culture plate, tomorrow I will start seeing colonies, right? So that is how they are there. Everywhere they are there. So they are cosmopolitan in nature, correct? Now, what else do you know about bacteria? Tell me in the chat box, all of you. What are these people talking about? Your JEE exam. First you write NEET exam properly. All nonsense. Yes. Okay, these people, students, these people can survive in extreme conditions. Okay. These people can survive in extreme conditions. That is, be it high temperature. Okay, be it high temperature. That means bacteria is present everywhere where other people, human life cannot survive, right? Extreme temperature, when the temperature is extremely low, okay, extremely low. They can survive in acidic medium. They can survive in salty medium, okay, saline conditions. All these conditions, these people can survive. Why? Because of the main reason is that these people have a cell wall. What is the cell wall made up of? Very important. Their cell wall, okay, their cell wall is made up of polysaccharide and amino sugars that they wrote. Okay, polysaccharide. Have we reached the target? Okay, amino acids, or else what did I tell you? Their cell wall is made up of peptidoglycan. Okay, peptidoglycan. And this peptidoglycan is so, okay, so good that they can survive in any condition. Okay, any condition. Got it? Neelam, what are you talking? Neelam is very busy. Very, very busy. Okay. Yes, peptidoglycan. And this peptidoglycan, another term that I told you was what? Murin. Okay, murin. Peptidoglycan, other word we tell is murin. That is, this, they are able to be, okay, they are able to be extremophilous because why? They can survive in all of them. Okay. Yes, students, we haven't reached the target, which is very, very disappointing. Very, very disappointing. Okay, fine. Now understand one thing. Monera's classification is there, no? So Monera is classified. Monera is classified into Archaebacteria, Archaebacteria, okay, and U-bacteria. Archaebacteria and U-bacteria. From the word itself, you understand, no? Archaebacteria and U-bacteria. U-bacteria means what? Ma'am, they are true bacteria, right? So this you remember, okay? They're true bacteria. Archaebacteria and U-bacteria. This is the classification. This is the classification. Very good. Yes. Perfect. Good job. Okay, now we will learn about Archaebacteria. Okay, before that, yes, shapes of bacteria. So students, bacteria are of different shapes. So depending on the shape, we have categorized them into 4 categories. What is the first category, ma'am? Spherical shape, or they are called as coccus shaped. Okay, cocci we tell, no? So cocci shape. Rod shaped or bacillus. Okay, lactobacillus. Okay, streptococcus pneumoniae. So I'll write, okay, I'll write whichever, like it is there in your NCERT, that I'll write. Streptococcus. That means it is spherical. Pneumonia, which causes pneumonia. Griffith also introduced us to Streptococcus pneumoniae, right? Now, rod-shaped bacillus, where do we know? Ma'am, lactobacillus. Right? Lactobacillus. That means these people are rod-shaped. Okay, rod-shaped. Then we have comma-shaped, that is Vibrio. Okay, Vibrio. And we have spiral shape, which is Spirulum. So you should say, know cocci, bacillus, Vibrio, Spirulum. You should know the scientific name. You cannot just write, okay, spherical, okay, rod-shaped. Yes? Tell me. Okay, good. Yes, very good. Coccus, bacillus, Vibrio, that means comma shape, Spirulum, that is spiral shape. So this classification is done based on their shape. Okay, based on their shape. Now, very, very important. Some additional points for all of you. Like this will be important for you. Okay, just it's easy also. It is prokaryotic cell. Obviously, Monera is prokaryote. Genetic material lies in the cytoplasm known as a nucleus. That means I told you they do not have a well-defined boundary, right? There is no nuclear membrane. Since they don't have nuclear membrane, the genetic material is going to be in that center, that is going to be called your nucleus. Cell envelope consists of glycocalyx, cell wall and cell membrane, which you will be studying in your cell unit, right? Glycocalyx, what is it made up of? The cell wall, what is it made up of? How does the entry of substance happen? All this you will study in cell chapter. Now, peptidoglycan, also called as murin, forms structural network of the cell wall, which makes them to be able to survive in extreme conditions, right? Which makes them to survive in extreme conditions. Are you all understanding? Extreme conditions. Now, peptidoglycan has repeating units of important, okay, N-acetyl glucosamine and N-acetyl muramic acid, which we will also call it as NAG, okay, NAG and NAM. In chemistry, you would have studied, no? NAG and NAM. N-acetyl glucosamine, N-acetyl N-acetyl muramic acid. So that's why murin word also comes, no? So it's easier. N-acetyl glucosamine, N-acetyl muramic acid. It's easy to tell also, no? NAG and NAM is there, which makes these people survive in extreme conditions, okay? 70S ribosomes are present in them. So 80S, 70S, you know, no? 80S we tell for eukaryotes, 70S we tell for prokaryotes. But again, it is your Swedberg's unit. It is not depending on the size or anything, because the smaller subunits are going to be divided into 50, 30. So it's going to be different. Understood? So 70S is going to be present. Now, nutrition and their metabolism. Now, nutrition means what? Mode of nutrition. As I already told you, what is mode of nutrition here? Yeah, NAG and NAM. Okay. Nutrition, as I told you, these people can do autotrophism. Okay. Autotrophism is there. Under autotrophism, I told you two things that they can do. What are they going to do, students? They can do normal photosynthesis. Okay, photosynthetic. That means in the presence of light. Okay, with the presence of light, they will make their own food. Or let's say yield energy. They can do this. Yes, ma'am. Generally, these people do this, so it's fine. Right? Generally, these people do this, so it's fine. Next one is the difference. What is that? Chemosynthetic. Chemosynthetic. Chemosynthetic means what? With the help of chemicals. Okay, utilizing chemicals. Okay, that is specially, students, these people are going to use what? They are going to use like your nitrates, nitrites. Okay, they are going to yield energy. They are going to yield energy. And with this, they also recycle. Okay, with this, they also recycle some other minerals like your nitrogen, phosphorus, sulfur, potassium. All these also they will recycle. What will this help, students? Obviously, ma'am, if they are going to recycle such kind of minerals, these are going to increase the soil fertility. So these bacteria are actually good for the soil, right? They are taking nitrates and nitrites from the atmosphere, fixing it to ammonia and yielding them energy. Okay, so I will write here. They are also going to give ammonia plus energy which ultimately is going to increase the fertility of the soil, just like how you have legumes. Okay, legumes. Okay, even yes, Fe also, even iron. Okay, all these they will recycle. Correct? Any doubts in this so far? Tell me in the chat box. No, right? It is easy, to understand? Yes, very good. So, photosynthetic light, okay, normally it will utilize light, photons, that is light, and they will yield energy. But here it is chemosynthetic, that means they are going to utilize, or they are going to take the help of chemicals. Okay, they are going to take the help of chemicals. With the help of chemicals, they are going to yield ATP or energy, and they will also recycle some of the other minerals by increasing the soil. This will increase the soil fertility. This is done. Now students, metabolism, when I talk about, understand that these people have simple structure. Okay, structure is simple, but their behavior, okay, or their metabolism is complex. Metabolism is complex. Ma'am, why do you say so? Because we have some examples, right? We have learned some examples in your biotechnology, how these people, as a part of their self-defense, okay, as a part of their self-defense, they make what, students? They do methylation. Okay, they do methylation. And they also, during their self-defense, ended up making endonucleases. Endonucleases. Not only that, when we learned about your Bt toxin also, from where this Bt toxin is coming, it is bacteria's self-defense to release Bt toxin so that this nematode does not affect me, or this pest does not affect me, right? So this bacteria is a very, very complex metabolism they have. They have the ability to do that, and they do this self-defense for whom? When a bacteriophage, okay, when a bacteriophage is affecting them, okay, a virus that infects bacteria is affecting them, that is when they do this self-defense. Yes or no? Any doubts in this? Because of that, what are they going to do, students? They are going to do methylation, and they are going to also do endonucleases, because of which the virus will get confused. Oh no, and their genetic material will not be able to express inside the bacteria. Okay, fine, good job. Okay, next one, see the structure is here. Okay, these are again finger-like structures. They are mainly used for movement. No, they are used for movement, but mainly they are used for anchorage. Okay, that means to attach somewhere. For anchorage, these people will be used mainly, okay, to the surface. Then you have ribosome, cytoplasm, plasma membrane, cell wall, capsule is there. Okay, this capsule also helps them to be very, very extreme. Okay, helps them to survive any kind of condition. The capsule also helps them to survive any kind of condition. Bacterial flagellum is there. Again, nucleoid is there, which is your circular DNA. Okay, now, Archaebacteria. Ready all of you? Not boring, right? Interesting, no? Chapter is interesting. Students, this chapter I know you might have studied with me only 3 times or 2 times, but then it is difficult to remember everything. It is very difficult to remember everything. Even before coming to the class, I revised the whole chapter. I read through the NCERT to come here, because even though how many times I am teaching, it is still important for you to revise. Okay? Yes. Okay. Now look here, Archaebacteria. Now, Archaebacteria, very importantly, you should know one thing. These people are extreme people. That means they can survive in extreme conditions. Bacteria itself can survive in extreme condition. Again, these people, they have given us some specific, categories. So, we are going to write them in extreme conditions. Now, ma'am, what exactly is the extreme condition they will be surviving? Students, they are thermoacidophils. Okay, thermoacidophils. That means what? They can survive in extreme temperature, okay, extremely high temperature plus acidic condition. Can you survive? Can you survive? Of course not. We cannot even think of surviving like that, right? Extreme condition, extreme high temperature and acidity, okay, thermoacidophils. Next thing what? They are halophils. That means, ma'am, these people can survive in extremely salty condition, like a brine solution, right? We tell extreme salty condition, saline condition. They can survive in extremely salty condition where you have rich NaCl. Okay, NaCl rich environment. I'll write that. Okay, NaCl rich environment. Then the last one we notice that these people are methanogens. Okay, that means they are present in the gut of ruminant animals and they can release methane, which is used for making the biogas. So they are methanogens also. That means they are present in the gut of ruminant animals. That is your cattle. Okay, ruminant animals, and these people are also going to help. Methanogens are also going to help in the formation of biogas. Okay, formation of biogas. Perfect. Now you can ask me a question. Ma'am, why this fellow is so extreme? Why can he survive in all extreme conditions? I am not even able to be in this 19 degrees Celsius. Why this fellow is able to do that? You can ask me the question, right? I obviously have answer for it. Archaebacteria's extreme condition, that means their survival to extreme condition, is because of the presence of pseudomurine. Pseudomurine. That means what, students? Cell wall is there, but it is false cell wall. Very good. It is false cell wall, right? Yes. Ma'am, one more reason could be there. Yes, because of the branched lipids. Okay, because of the presence of branched lipids. Ma'am, could there be one more reason? Yes, students, there is one more reason, that is because of the ether linkage. You have how ester linkages are there, no? Because of the presence of ether linkage. Because of these three things in their structural modification, they are able to survive in extreme conditions. What is it? What is it? What are they telling? Okay, some famous people in the chat box, huh? Okay, so here it is because of the presence of pseudomurine, that is false murine. Murine, I already told you, peptidoglycan. And then it is because of the presence of branched lipids, also presence of ether linkage. Done? Thumbs up in the chat if you understood this. So here you can see, students, halophiles. Okay, here you can see thermoacidophils. Most of the structures you will see, they are bacillus. Okay, rod shaped structures. Done? No doubts in this? Okay, good job. Okay, good job. So this is your Archaebacteria. That means they will be able to survive in extreme condition. And three categories we have: thermoacidophils, halophils, and methanogens. Methanogens is present in the gut of the ruminant animals like your cow and buffaloes, and that will be used for biogas because they can produce methane. Okay, halophils, NaCl rich, that is salty condition, and thermoacidophils, temperature will be high. At the same time, high acidic condition also, these people will be able to survive. And the three reasons for their survival is that. Okay, three reasons for their survival is that. Okay, next is Eubacteria. Any doubts in Archaebacteria? Anything did we miss? No. Okay, very good. Done, done. Okay, let's do Eubacteria. Yes. So Eubacteria, from the name itself, I know these people are true bacteria, ma'am. Right, true people, true students means the ones who always attend the live, give us the comments, so encouragement. True bacteria. True bacteria is also because of the presence of peptidoglycan. They do not have pseudomurine. Okay, they have murine. Peptidoglycan, or they have murine. Yes. What else do you know about Eubacteria? All of you give me 1 points about Eubacteria. Let's see how many of you have revised something. Okay. These people, as I told you, they are true bacteria, students. They have rigid, okay, they have rigid cell wall. Rigid cell wall is present. Okay, ma'am, understood. Now, Eubacteria can be categorized. To make you understand more in concept, they have to be categorized, okay. So, Eubacteria is further categorized into cyanobacteria, and mycoplasma. Now, cyanobacteria, cyan is a color, student. C-Y-A-N, color is blue. Okay, cyan is a color, blue. So I can call this also as BGA. I can already see some of you telling in the chat box, blue green algae. I can call it as blue green algae. Ma'am, algae? That's why I told you some confusions have been. That is, some things we study here, some things we study further. Okay, yes, correct. Okay, algae, that is your blue-green algae, and then you have mycoplasma, which is the smallest living fossil known, smallest living cell known. Okay, now this cyanobacteria, very, very important to understand, they can be present. They are present, ma'am. They can be present as colonies. Okay, colonies. They can be present individually or they can be present as colonies. Got it? Colonies means what? Ma'am, like many, same maybe cultured people, same caste people, have you seen they make colonies and stay together? Or they can also be present exceptionally filamentous. Okay, filamentous. How is filamentous? Okay, how is filamentous? I will show you here. Okay, how is filamentous? I will show you here. Yes. Now, filamentous you have to know because the structure is given in your NCERT. Okay, 1 is there, 2 is there, 3 is there. They won't be in straight line and all. Okay. Okay. This is their structure. Okay, this is their so sorry, students. Okay, I laughed at my own diagram because it's too. Okay, yes, better. Now, very, very important. This structure, okay, this structure is covered by a mucilaginous, okay, mucilaginous or a gelatinous sheath. They're covered by what? A mucilaginous gelatinous sheath. Like this one sheath is covering them. Why, ma'am? It's covering them for protecting. Okay, I'm laughing looking at my diagram. Okay, first when I drew, I felt like I should laugh. Why I can't laugh? Only you people can laugh. Okay, this is a mucilaginous. Okay, mucilaginous, or we can tell gelatinous sheath. Students, why, ma'am, you have written both the words? You can just write one word because your NCERT has given both the words. If you see your NCERT properly, in the paragraph one word is given, on the diagram one word is given. But when I searched on what further is the difference or is there any difference, I realized that mucilaginous is a glycoprotein. Mucin, remember, I taught you in structural organization. So mucin is a glycoprotein, but gelatinous sheath is a protein. Okay, it's a protein, that is collagen. So it's a protein. Okay, so that is what we understand. One is a glycoprotein, one is a protein. Okay, that's all is the difference because, okay, because both are written in your textbook, you can write both of the words. No one is going to cut your marks because of that. Okay, no one is going to cut your marks because of that. Now, students, if you look at the structure, there are some specialized cells in the middle. Okay, there are some specialized cells in the middle, which I'm drawing now for all of you. Okay, yes. Yes, can you see some specialized cells I have drawn pink color? Yes, yes. Okay. Yes, very good. The specialized cells, okay, those specialized cells are called nothing but heterocysts. Those specialized cells are called nothing but heterocysts. Okay, heterocysts. I hope those words come back. Now, what is heterocyst? These are special cells, okay? These are special cells which are going to do, okay? Special cells which are going to do, one second, yeah. Special cells which are going to do nitrogen, oh God. Going to do nitrogen fixation. Nitrogen fixation. Okay, special cells which are going to do nitrogen fixation. One thing is very important, these cells should not have any oxygen coming nearby, because oxygen will react with nitrogen very fast. Okay, so nitrogen fixation time, this oxygen should be avoided. That is why you have this mucilaginous coat as well. They will do the protection of oxygen not entering, otherwise nitrogen will go and combine with oxygen. Okay, because oxygen is very reactive, it will go everywhere, go combine everywhere. So heterocyst is specialized cells which is going to do nitrogen fixation. Now here the example given in your NCRT is going to be your nostoc, okay, nostoc and Annabina. Don't write Annabella, okay? Annabina is going to be there. Okay, nostoc and Annabina, which will stay as your filamentous structures, and these people are capable of doing nitrogen fixation. Again, some symbiotic association also is there, like your Azola and Annabina. Like that's different examples are given. Okay, yes, did we reach the target? Oh yes, oxygen always ready to mingle. Okay students, these people, this blue-green algae are the main reason for algal bloom. Okay, they make this algal bloom, and that time they will have gas vacuoles. Okay, they'll have presence of gas vacuoles, and these people do algal bloom. Algal bloom, I hope all of you know, like if you have seen a lake which is not used by people for a very long time, you will see that green color patch like structure on top of that. That is called algal bloom. Now what they will do, they will not allow the aquatic organism to breathe, or they will not give any dissolved oxygen for the aquatic organism. They will utilize all the oxygen. They are like very selfless. Okay, they will just utilize all the oxygen and not give anything for the aquatic organisms, which is very dangerous. Okay, which is very dangerous. Perfect. Just check. Yes, good job. Okay, now this blue-green algae, students, has one more thing. These people have the presence of pigment. Okay, whatever is exception, like special characters I'm writing with orange color, they have pigments which is called as chlorophyll A. Okay, chlorophyll A pigments are present just like in higher plants, which will help them to do photosynthesis. Okay, which will help them to do photosynthesis. Understand, majority are not autotrophic. Again, some of them are autotrophic, some of them are heterotrophic again in bacteria. So remember that, some are autotrophs. Okay, some are autotrophs, and majority are heterotrophs, because everyone need not have the pigment, right? Majority are heterotrophs. Got it? Clear? Exceptional points also I have written. Perfect. Okay, did we miss something? Mycoplasma we should write. Mycoplasma I'll teach you. Okay, after some time. Now, reproduction in bacteria. Reproduction in bacteria. Again, I told you symbiotic association, Azola is given, so that you just read from there. Okay. Now, reproduction in bacteria. These people are very simple organisms. Their first mode of reproduction is going to be with the help of binary fission. Okay, binary fission. But when the condition becomes unfavorable, these people will immediately go for what? They will go for spore formation. Okay, they will go for spore formation. When the condition becomes unfavorable, they'll form spore, and when the condition becomes favorable, they will break open the spore and release the spore. Okay, but then students, because every other organism around them is doing sexual reproduction, bacteria also felt bad, so bacteria will also do sexual reproduction. But you cannot tell, you know, like you cannot tell like advanced or anything like that, but understand, bacteria also over time has evolved. If evolution should happen, if variation should happen, sexual reproduction should happen. So bacteria went to his parents and asked, do we do sexual reproduction? How did I become from this to this? Okay, how am I capable of doing this? So the parents told her, yes, we do sexual reproduction in the form of conjugation. Okay, we do sexual reproduction, but we do not do like other, okay, the silly organisms, but we do in a better scale, that is called as conjugation. Very good. We do conjugation Okay, that means a small portion of DNA transfer happened. We can also write with conjugation, you can also write transduction. Okay, transduction and transformation. Okay, transformation like we studied in Griffith's experiment, no? Streptococcus pneumoniae was able to do transformation. If they were not capable of doing that, then how can, our strain become virulent? Okay. From Griffith's experiment, we learned that they can do transformation, right? They can do transformation. Yes, we reached the target. Good job. Okay, so conjugation, transduction, transformation, and conjugation, transduction, and transformation. That means, students, a small portion of the DNA. Let's say this is your one strain. Okay, this is one strain. From this strain to this strain, a small portion of the genetic material is getting transformed. Okay, small genetic material is getting transformed. Clear? No, cockroach. What evolution in cockroach has happened is because of human intervention, no, anthropogenic activities. They have got resistant to most of the chemicals that we are using now. That is a kind of evolution. A small variation has happened over generation. Yes, so this is what it is. Okay. Clear, students? Binary fission, okay, unfavorable conditions. So again, binary fission is this what is happening, okay? This is the cell, right? This is the cell, partitioning will happen. Again, one daughter cell, one daughter cell. Here what is going to happen? Spore formation is going to happen. Spore will go out, okay? Spore will go out when the condition becomes favorable. That's it. That's it. Okay, perfect. Now mycoplasma will study. Yes, mycoplasma, okay, mycoplasma, remember, completely lacks a cell wall. Okay, they do not have cell wall. Smallest living cell known and can survive without oxygen. That means these people are facultative. Okay, these people are facultative anaerobes. That is, oh ma'am, oxygen is there, I will breathe. Okay, anaerobes. What is the meaning of that, students? Ma'am, you're teaching, okay, I'll sit in your class. Ma'am, you're not teaching, okay, I will not learn. Facultative, that means full. If oxygen is there, okay, I will breathe with oxygen. Oxygen is not there, okay, fine, I will not breathe with oxygen. So that kind of people, facultative anaerobes. Many mycoplasma are pathogenic in animals and plants. And remember, students, these people causes. Okay, this is a disease, okay. Phylloidy is a disease where the flowers, okay, the flowers will be replaced by leafy structures. Replaced by leafy structures. This can make the plant sterile. Okay, this can make the plant sterile. It mainly affects strawberries, roses, etc. Fine? Okay, yes. Yes, very good, Raju. These people are also obligate parasites. They can cause pathogenic effect on plants and animals. Okay. Now, here, students, I have just put this box for you to highlight one thing. This one. Okay. Archaebacteria is ether linkage and these people have ester linkage. So, these were some of the differences also Carl Woese would have studied. Okay. That is about the lipid branches and other things. Done. Can we go to Protista? One kingdom we have done and dusted. What did we study? We studied everything about Archaebacteria and U-bacteria. We also studied about Monera, how they are. Their sole members are bacteria. These people can stay in extreme condition, be the temperature high, be the temperature low, be acidic condition, be alkaline condition, methanogens inside the gut of ruminant animals. They will be able to survive every single place. Okay. Archaebacteria extremophilous. That means they have thermoacidophils, halophils, methanogens, and these Archaebacteria have pseudomurine, that is false murine or false peptidoglycan, because of which they are able to sustain in all this extreme condition. Also the presence of branched lipids, also the presence of pseudomurine, branched lipid, and also they have structures which are capable, like in the sense they can withstand, okay, they can withstand, and additionally they have ether linkage, okay. The eubacteria have ester linkage, these people have ether linkage. I think you people know chemistry better than me, okay. Then students, we studied about eubacteria. Eubacteria have cyanobacteria as well as mycoplasma. Cyanobacteria is called as blue-green algae. These people have chlorophyll pigment in them, chlorophyll A. These cyanobacteria can be living alone, okay, or in colonies or in filamentous structures. In that filamentous structures we have studied, and they have specialized cells called heterocysts which are able to do nitrogen fixation. These nitrogen fixation will help to increase the soil fertility, to make the environment more efficient for plants to grow. Why? Because they are going to take the atmospheric nitrogen, convert it into ammonia and fix it in the soil. That's why we keep legumes, right? Pea plant, bean plant, we keep between the rice crops. Why? To increase the fertility of the soil. Got it? Yes. Fertility of the soil. Okay. Done? No doubt so far, I will. Yes, I just drank water. Okay. Yes, no doubts, I hope. Perfect. Okay, next one we are going to learn Protista. Okay, we are going to learn Protista. Now students, Protista, I have very few things to tell, so I will write it on this slide only. You have kingdom Protista. Now Protista's main thing is, these people do not have clear boundaries, just like we just explained right now, no, that these people you know, have amoeba, have paramecium, okay, but these are doing some kind of locomotion, no problem in that. They do not have proper boundaries, and because of it there are some categories of them which are very plant like, some of them are fungi like, some of them are animal like. So this is why we tell that Protista does not have boundaries, okay, they do not have clear boundaries. Clear boundaries. They do not have clear boundaries, okay, no clear boundaries, and they are a link, I can say they are a link between plants, animals and fungi. I'll tell you the reason also, okay, reason also I'll tell you. Yes, very good. Now look here students, they are divided, okay, kingdom Protista is divided into cryophytes, dinoflagellates, euglenoids, slime molds and protozoans. Under protozoans you have amoeboid, flagellated, ciliated and sporozoans. Now if you look here, cryophytes, phytes, bryophytes, pteridophytes, so that phytes word, no, so here cryophytes, these people are capable of doing autotrophism. So this category, okay, these three people, these three will come under your plants, more plant-like, okay, plant-like, very good, they will be more plant-like because they are able to make their own food. But when you come to slime molds, you will notice that these people are more fungi-like, they cannot make their own food, and these people do your spores, formation of the spores. Got it? Yes or no? Okay. Protozoans on the other side, they are more animal-like, okay, animal-like. Done, done, done. Are you resigning alarm posting? Oh, don't tease, okay? You people are not capable of doing, no? Then why are you teasing other person? Came off, big people. Oh. Yes. Yes, and here when we talk about, understand, the main thing that we are going to study about these people are going to be about their cell walls, because question can be purely based on the presence and absence of cell wall, and some of them have cell wall present in some, absent in some. We are going to study the different types of cell wall, or what is not there. Instead of cell wall, what do they have? Okay, instead of cell wall, what do they have? Yes. Animal character because of their locomotion, okay, presence of locomotory structures. Okay, let's continue. Students, I'm only going to write the main characters here. Okay, I'm going to write the main characters here. Okay, aquatic, floating, all that I will not be writing. I will just write the main characters. So here, chrysophytes. As I told you, these people are more going to be plant-like. Okay, they are going to be more plant-like because of the presence of chlorophyll and able to do autotrophism. Main two people who are going to fall under this is desmids and diatoms. Desmids can also be called what? Desmids can also be called as your golden algae. Golden algae. Now, diatoms is the main part that you need to study. Okay, main part that you need to study. Yes, here students, diatoms have cell wall. Okay, they have cell wall, but their cell wall is overlapping. Okay, they have overlapping shells which are present in the form of soap box like. That means what? One box is like this, one box is like this. Remember, we used to keep soap like this, no, before. Nowadays, we have all those attached ones, but before, all of you remember, we used to go to shop and get green color, orange color. The down will be a circular, oval shaped one, and top will be a box shape. So, we used to close and keep. Okay, I think now also some of you will have at home where you keep the washing soap. Okay, that is a soap box like. That means overlapping structures, overlapping shell like structures. These are called as your soap box like structures. Okay, these are your soap box like structures. Soap box like overlapping shells are present. Okay, what is this overlapping shells made up of? This overlapping shells are made up of silica. Okay, silica. And students, this silica is indestructible. That means they are very, very rigid. They will not be able to destruct easily. Now, what is going to happen? Okay, what is going to happen? Very importantly, they are going to happen is that these diatoms, okay, these diatoms, that is these shells, no, will continuously deposit. What they will do? They will continuously deposit on the earth bed. And students, after millions of years, these people are going to make a layer called as the diatomaceous earth. Okay, diatomaceous earth. What are they going to form? Okay, diatomaceous earth. What is this diatomaceous earth rich of? Ma'am, these people are rich. Okay, these people are rich with the silica. Okay, they are rich in silica. And what will they be used, students? After millions of years, these things are going to be used for filtration, polishing, etc. These will be used for. And these people are the chief producers in the ocean. Okay, chief producers in the ocean. Any doubts so far? Tell me. Yes, polishing, filtration of oil. Very good. These people are going to be what? These people are going to be chief producers, and this has come as a PYQ in the past years. These people are chief producers in ocean. Okay, chief producers in ocean. That's it. Very simple. Cell wall is there, but their cell wall is made up of overlapping shell that is made up of silica, soap box like structure. Silica will continuously deposit over the years and make an entire bed called as the diatomaceous earth, which is rich in silica, which is used for polishing and filtering. Okay, polishing and filtering. Correct? Awesome, awesome. This is your plant-like desmids, diatoms. These people also, I'm telling they are plant-like because they are able to do autotrophism. Okay, autotrophism. From the name itself, you understand, no? Chrysophytes, like bryophytes, pteridophytes. Okay? No. Only desmids are called as golden algae. Okay, only desmids are called as golden algae. Please don't confuse yourself. Okay. Yes, they are marine. Obviously, they are marine. They'll be floating. Okay, those things you can general study, but questions will be based on the different characteristics. They will not ask you if they are floating or not, because why will they? Okay. Now, dinoflagellates. Okay, dinoflagellates. Now, students, these people are also mostly marine. Dinoflagellates. That means, ma'am, these people have something to do with flagella. Yes or no? Yes, of course, they have something to do with flagella. That is, students, okay, I'm going to draw a very structure. Okay, dinoflagellate is there. This dinoflagellate, okay, dinoflagellate is going to have flagella. Dinoflagellate, so two flagellas they will have. One is going to be a transverse flagella, one is going to be a longitudinal flagella. One is going to be a transverse, I think the color is not visible, no, 1 second. Okay, one is going to be transverse, one is going to be longitudinal. One is going to be transverse, one is going to be longitudinal. So what is going to happen, students? They are going to have two flagellas, one transverse, one longitudinal. Okay. And both of them are going to arise from a furrow. Okay, a furrow kind of a structure. Both of them going to arise from a furrow. That's what I drew there. Okay, yes. Very good. One transverse, one longitudinal. Yes. Christiana, what are you here for? Okay. Yes. Is this clear? Flagellate is clear? Okay, you can either draw it upside or you can draw it downwards. Okay, one longitudinal, one transverse. Now, students, these dinoflagellates are present in most of the colors. Okay, they are present in most of the colors like red, okay, yellow, brown, green, blue. Okay, they are present in most of the colors. Now, what you have to remember, I told you that the main question that is going to have, very good, who answered that in the chat box? Neelam, yes, Neelam is back answering, yes. Furrow is just a small, furrow means what, like a small depression through which the flagella is going to come out, okay? Yes, very good. They are going to appear in yellow, green, brown, blue or red, depending on what, students? Obviously, depending on their pigment, right? Depending on their pigment. Depending on their pigment. Very good. Now, very important, students, understand, these people have stiff cellulosic plate depending on the main pigment. Okay, whichever pigment is present, depending on the main pigment. Yes, as I told you, these people are going to have stiff cellulosic plate. Stiff cellulosic plate, which is going to act as their cell wall, correct? Yes, ma'am. Okay, they will have a cell wall which is a stiff cellulosic plate on the outer surface. Now, students, most important example that is given in your textbook is the presence of Goniolex. Okay, they have spoke about Goniolex. So, here an example that they gave is about Goniolex. Okay, Goniolex. What have they told about Goniolex? All of you tell me in the chat box. Yes, tell me in the chat box. Quickly. Someone is telling, Goniolex, who told? I think Roshni has told. Okay, Goniolex. Now, what happened in Goniolex, students? These people can do, they did rapid multiplication. Okay, they underwent rapid multiplication. And what did they do? They gave the appearance of red tide. These are red colored, okay, dinoflagellates. These people, because of rapid multiplication, gave the appearance of a red tide. okay, red tide. But the problem is, yes, it looks beautiful to my eyes, but it is not that beautiful. Why? Because of the presence of this Goniolex continuously releasing a toxin. Okay, they continuously release a toxin called as saxitoxin, which kills other aquatic organisms. Okay, which kills other aquatic organisms. Is this okay? Yes, it might look beautiful. You will see a red color tide coming. It looks very beautiful, ma'am. In a blue, okay, in a blue waters, okay, in blue waters, let's say blue waters, if you see a red tide, obviously you will be excited. Wow, one red tide is coming. Beautiful it will look. But that beauty is not going to be, okay, the beauty is not going to be lasting for longer, because other aquatic organisms in that ocean, in that sea, is going to die because of the release of saxitoxin or toxin, which is mentioned in your NCERT, that will kill other aquatic organisms. Okay, perfect. Yes, good. Kills other toxin, other organisms. Okay. Okay. Clear. This is perfectly clear, I hope. Good job. This is what a major portion that is given in your NCERT. Okay. Dinoflagellates is done. Yes, ma'am. Now, next is euglenoids. What is euglenoids? You know euglena, right? Euglena. Now, this euglenoids also are going to be plant-like. Okay, they are going to be plant-like, but they have a problem. The problem is that, students, in the presence of sunlight, they will do autotrophism. They will do what? Autotrophism. They will make their own food. But in the absence of sunlight, okay, absence of sunlight, what will they do? They will become heterotrophs. They will become heterotrophs. What will they feed on? They are only smaller organisms, but they will feed on more smaller organisms. So let's take an example. Okay, now Kubera, Meghana, Sanvi, Nagaraj, okay, Raju, all of you went for a party with Euglena. Okay, you people are all smaller organisms. Okay, what Euglena will do? When sun is there, Euglena is sunbathing. Euglena is making autotroph, its own food it is making. But suddenly when the sun goes, it will start eating you people. Smaller organism, it will start feeding. Can you imagine? So Euglena has these two characteristics, that is, in the presence of sunlight, they will do autotrophism. They'll make their own food. In the absence of sunlight, they will suddenly start feeding on smaller organisms. So all of them who went for tea party will not be there after the party. They would have vanished. Why? Because Euglena would have consume them. Understood? So this is one of the specialized characteristics of Euglena. Now students, these people do not have cell wall. Okay, no cell wall, but instead of cell wall, okay, in the absence of cell wall, they have a protein. Okay, they have a protein rich flexible layer called the pellicle. Pellicle. Correct? Yes, ma'am. Of course. Pellicle. Yes. So, since these people are doing this two kind of category, I will call it mixotropic. Okay, mixotropic. That means they are doing autotrophism as well as heterotrophism. So mixotrophism. Okay, so we can call it mixotrophic. These people have no cell wall. They have protein-rich flexible layer called the pellicle. Okay, that also no one has it out. Now, students, these people also have flagella, but their flagella, what is going to happen? Okay, their flagella, one is going to be short. Okay, one is going to be short, one is going to be long. Okay, so they will have two flagella, here also two flagella, but one short and one long. One short, one long. Yes, good. Okay, most of it all mixotrophic when I was teaching the last one. So I was thinking what happened to you people, but mixotrophic here you will tell. Correct? Yes, awesome. Okay. Yes. Now here students, one more PYQ question you have, that is Euglena has a pigment, okay, like higher plants. What is that pigment is the question. So Euglena and higher plants, okay, Euglena and higher plants, okay, they have one pigment. Which is that pigment? Chlorophyll A. Okay, chlorophyll A is the pigment that they have. That's it. Okay, pigment that they have is chlorophyll A. Perfect. All the main characteristics we have discussed. Okay, next is going to be a slime mold. For slime mold, I have put more images for you to make you understand. So look at this. They don't have chlorophyll. They are surrounded by plasma membrane only. However, their spores have cellulose plate. So the spores will have cellulose, but the organism itself will not have the cellulose membrane. Okay. Saprophytic nutrition. Slime mold resembles both protozoa and a true fungi. Why? They are like protozoa in amoeboid plasmodial stage, and they are similar to fungi in spore formation. So these people, okay, these people are going to show two things. What is that? They are like protozoans in amoeboid plasmodial stage. That means these people will scatter, okay, and they will grow. That time they are going to act like a protozoan. But when it comes to spore formation, they are going to be a true fungi. Okay, they are going to be a true fungi. Thank you, Delvi. So here students, see, under suitable condition, they form an aggregation called as the plasmodium. All of you remember? This is what you are seeing, okay, plasmodium. That means what they will do, students, like this one aggregation, they will form during the aggregation, okay, during the aggregation, they will form plasmodium. Like this structure, plasmodium they will form. That is, when the condition got unfavorable for them, they will wait. That okay, let the condition become favorable, then they will go. The mature plasmodium forms young sporangium. The mature sporangium will bear spores. The spores undergo meiosis. Okay. The mature sporangium burst open and release the spores. The spores possesses true walls. Very important. Those people did not have wall. Here they have true walls, and they will have extremely resistant and survive for many years. The spores are disappeared by air currents. The spores germinate and differentiate into flagellated and amoeboid spores. During this time, they are going to act like the protozoans. Clear? Yes. Flagellated and amoeboid spores fuse together to syngamy and forms the zygote. Zygote again develops into mature plasmodium. Don't have to take tension. What is that? Very simple things they have mentioned. Yes, ma'am. That is, they will form plasmodium. They will form plasmodium, which is an aggregate. Okay, aggregate. Now this will mature. It will mature. And when it matures, it will release the spores. The spores will go with the help of air currents, and the spores will later form your amoeboid and flagellated. Two different types of spores will be formed. Okay, two different types of spores will be formed, amoeboid and flagellated. They both will fuse. Okay, amoeboid and flagellated. These both will fuse, and what will happen, students? They will form the zygote. That's it. That's it. Can you remember? Is it difficult? Is it difficult or easy? Mainly two things you people have to remember. The spores will have true walls. Okay, remember that. The spores will have true walls. Why? Because they have to survive in all the resistant environment, right? They have to survive for a longer time. They have to be carried away by the air currents. So it's not an easy job. It is not an easy job. Yes. Good job. No doubts, no? Nothing. Just remember plasmodium and aggregate. When the condition becomes favorable, these people will mature and release the spores. Spores will have true walls because they have to be resistant to all the conditions. That will be taken up by the air currents. In that, they are going to form two types of spores, amoeboid and flagellated. That's when they behave like a protozoan. Otherwise, they behave like a fungi. That's why I told you slime mold is very near to fungi. Okay, slime mold is very near to fungi, I told. Now, protozoans, students. Protozoans, we have mainly 4 categories. What are the main 4 categories? And these people are considered more animal-like. Okay, more animal-like because of the presence of, locomotory structures and other things. So the first one is amoeboid. Okay, Amoeboid protozoan. Second one is ciliated. Next one is flagellated. Okay. Protista and protozoan, don't get confused. Under protista, your protozoan is going to come. Okay. And last one is spore formation. Right. The sporozoans. From the name itself, you understand something. No, some hint you are getting. Okay. Hint you are getting. Doubt is not doubting. Okay, good. Okay, doubt shouldn't doubt. Unnecessarily, why will it doubt? Yes. Okay. Fine. Okay, we will move forward. Okay, we will move forward. So, students, here, first we will start with amoeboid protozoans. All of you tell me all the points you know. First of all, amoeba. What is amoeba? Ma'am, we have all studied amoeba. Even your parents will know amoeba because amoeba is pretty famous. Okay, amoeba is pretty famous. Everyone, when we talk about biology, the first thing they tell is, amoeba you study. They ask like this, amoeba you study. So, because parents only know amoeba, they talk about amoeba, right? So, amoeba is very famous. Now, why amoeba got famous? Because of the presence of their false feet. What is a false feet made up of? Pseudopodia. Pseudopodia. False foot, false feet. This false feet is going to help them in locomotion, capture and all of that, right? So when we talk about amoeboid protozoans also, we talk about pseudopodia, right? Yes. Yes, good job. Tell. Palak Chowdhury, you will get Palak. Yeah. You will get the notes in the description box after I finish the class. Okay? Because I'm writing the notes, no? How can I give the notes before only? Yes. So these people, students, these people are going to do the job of what and all? They are going to help with capture. They will engulf, right? We can use the word engulf. They will engulf their prey. They will also help with locomotion. Locomotion. They are going to be on the marine environment. Okay? Marine, free living. All this you know, no? Free living. Okay, marine free living. Now, very important is the examples. They are going to ask you the examples. Here, what is the examples? Entamoeba. Entamoeba histolytica. Where have we studied Entamoeba histolytica? Ma'am, in the second Biology 360 club only we studied. Entamoeba histolytica cause amoebiasis. Right? It causes amoebiasis. So, this is your amoeboid protozoans. Okay. Next one is flagellated. Now, students, here flagellated, obviously they have flagella, right? They have flagella. Whatever the structure be, they have a flagella. Now, flagellated protozoans also, here we also talk about mainly the flagella. Okay, flagella. These again help for locomotion, for anchorage. Okay, for anchorage. Helps to catch the prey. Catch the prey. Okay. Yes, good. 1 second, students. Okay. Correct. Very good. Very good. Example is important here. Trypanosoma, right? Sleeping sickness. These people also, students, what are they? They can be parasitic. They are parasitic. And you have example. Trypanosoma, which causes sleeping sickness. Which causes sleeping sickness. Done? Yes. Okay, done? Perfect. Now, next is ciliated paramecium. Okay, ciliated, here you have example paramecium. In paramecium, students, again, lot of examples are not there. These people, how do they look like? They look like the sole. Sole, no? Like your shoes. soul will be there. No, they look exactly like this. These people will have micronucleus, macronucleus. Now here only thing, active, okay, active movement happens with the help of active movement of food, everything, okay, of food happens, okay, active movement of food happens with gullet or, okay, this is a small opening. Can you see an oral groove? That oral groove is the gullet, okay. Yes, what, Palak? If you're so concerned, I think you shouldn't be here. Okay, so here these are oral groove, oral groove, okay. These, through this only the food and everything will enter and it will go inside, and you can see here the digestion will happen, and whatever is left out, they will also come outside, okay. These people will have micro and macro nucleus as well. More structures are there to these people, okay, but here in your NCERT only this is mentioned, correct. Here you have ciliated. Any doubts? Should I write that? Micro, macro, okay. Micronucleus and macronucleus. Okay. See, paramecium and amoeba, which we discussed there in animals, came here. Right? Yes, very good. Now, sporozoans also nothing, students, because these people are capable of doing spore formation. Okay, spore formation, or they have a let's say spore forming life also. Okay, spore forming stage, not life, spore forming stage in their life cycle. Like example, Plasmodium vivax. What does Plasmodium vivax do? Ma'am, they causes malaria. Okay, Plasmodium vivax, that causes malaria. That causes malaria. Correct? These people have a sporocytic stage. No, that means that sporozoites which you studied there is because of the spore forming stage. So if they are capable of forming spore, or they are having a spore forming stage, then these people will be, okay, these people will be coming under sporozoans. Sporozoans. Yes. Good job. Yes, Plasmodium. Here, Plasmodium vivax, you are talking about. Falciparum also is there, which causes malignant malaria. Okay, which causes malignant malaria. Okay, students, before fungi, I will just take a 5 minutes break. Okay, I'll just take a 5 minutes break and I'll be back. You people don't go anywhere. Okay. After that, fungi, and we will be done. Okay, after fungi, now it's 8, 9. Okay, 8:15. Yes. I will just get some hot water and come. So till that 5 minutes, you people wait here. Okay. Plasmodium vivax, dear. Yes. You people also drink some water and stay back here. Don't go anywhere. Yes, I'm back. Are you all back? Tell me in the chats. Okay, let's start with fungi, students. So fungi was put into a separate kingdom because of R.H. Whittaker. Why so, ma'am? Because these people are completely heterotrophic, right? Heterotrophic, and these people's heterotrophism is mainly saprophytic, and they can be, some of them are parasitic as well. We have learned about ringworm and all of that, no? Now these people cause pathogenic, they can become pathogenic as well. Now what else do we know about fungi? These people are also cosmopolitan in nature. That means they are present almost every single place. They are present every single place, and they show great diversity. They show great diversity. Correct. Now if you see here itself, you will see that bread mold is there. Okay, bread mold is there. Toadstools is there, rich source of vitamin B, that is your mushroom is there. Now there are some which are edible to us, there are some which are non-edible to us, right? All of you have seen? Yes? Very good. Now students, what happens? These people are loves. Okay, these people love one thing, that is moisture. Okay, they love to grow in place which is rich in moisture. Okay, they love to grow in places where there is moist environment. Now all of you would have seen, no? Normally what happens, you love bread on the first 2 days. Okay, first 2 days you love bread because you want to eat it with jam, you want to eat it with peanut butter, you want to eat it with some chicken curry. Okay, all that agraha you will have on the first day. But as days goes, second day also still you will have some interest. Third day you will forget that you bought bread and kept. What will happen? After a few days your mom will go and check, okay, that wherever you kept the bread, no, slowly on the white bread she will start seeing some green dots. Okay, she will start seeing some green dots, or by the time your mom saw you, she will already start seeing the black dots. Now what was exactly happening there? You will get hittings, that is so for sure. Okay, that cannot be avoided, it is not something that we can avoid. But on that later you will start seeing growth like this, and you will start noticing that there is some cotton like structure growing on this. Now what you exactly grew there is rhizopus. What you exactly grew there is rhizopus. Without your knowledge, you were growing rhizopus, but what did you get? The cost of rhizopus is one hitting on you. Correct? One hitting on you. So on the bread, students, what is growing is your rhizopus. Now if you see your rhizopus, they have a specialized structure. I will go into the structure, but right now tell me if you understood this. Okay, now students, these people, we cannot tell that all the fungi are bad, because why? These people also show symbiotic relationship. Symbiotic relationship. Ma'am, what is symbiotic relationship? That means, see, you're also benefited, I am also benefited. That means what? Fungi also is benefited, algae also is benefited, higher plants are also benefited, mycorrhiza also is benefited. This kind of an association. That means symbiotic relationship, you will study your lichens, where it's a relationship between algae and a fungi. Now what do these people benefit each other? Fungi will give protection to algae, and algae will give food for fungi, because fungi does not know to make food, right? Fungi does not know to make food. So algae will give food and fungi will give shelter. Fungi will give shelter. Correct? Now same relationship, it is there in higher plants as well, which is seen in, okay, I'll just erase this. Okay, yeah, it is seen in mycorrhiza, okay, and plants, higher plants. This is also one kind of symbiotic relationship. So I cannot say all the fungi are bad. There are fungi who are doing good also, that is, they are providing the necessary nutrition for the plant, because the plant is tall enough, no, it cannot reach, so the mycorrhiza is helping for that. The plant is giving protection to the fungi and also giving food for the fungi. This is the general understanding of fungi. Now students, two examples given specially in your NCERT is your Puccinia, that is in wheat rust, okay, wheat plant, the rust formation, and white spots that you see on mustard plant, okay, white spots that you see on mustard plant. Now that much is clear about fungi. Symbiotic relationship, heterotrophic, saprophytic, they can be pathogenic as well. Now, fungi structure is very important, students. Normally, see, when I drew the bread, okay, I can't copy paste the bread, otherwise I would have copy pasted the bread. Now, this is the bread, okay, sandwich bread we are taking, students. We are very professional, okay. In drawing, no, we don't do any kind of discount, right? Okay, this is the bread. Does it look like a bread? Yeah, okay, this is the bread that we drew. Okay, on that we saw first some green spots. Okay, some green spots we saw, and some green spots turned into black spots, and later those developed into your rhizopus. Okay, rhizopus. Now this is one of them. Okay, one of them. I'll draw one more. Now, structure of fungi is very, very important, students. That is, these people are made up of, okay, made up of slender, filamentous, okay, slender, filamentous. Main word that you have to use is thread-like, okay, thread-like structures called as what? Hyphae, ma'am, called as hyphae. Thread-like structures called hyphae. Now, many hyphae together, okay, many hyphae together will form a network called as mycelium. Okay, this is one hyphae, this is one hyphae, this is one hyphae, this is one hyphae, this is one hyphae. Many hyphae together will form a network. Okay, many hyphae together will form a network. So, a network of hyphae will be called mycelium, ma'am, called as mycelium. Done? Perfect? Easy? Yes, very good. Network of hyphae is called mycelium. Now, students, hyphae can be of two types: aseptate and septate. Aseptate and septate. Now, what is aseptate and septate? I will draw the diagram and show you. See. What do you look at it and understand? Ma'am, you have drawn a structure where there is multi-nucleated structure, but I do not see any barriers or boundaries. Okay. Now, let me draw this. Okay, I have drawn one more structure for you people. Okay, notes will be available on Biology by Gopika as well as Vedantu NEET English. Both the telegrams, the notes will be available. So, do not fail to join the telegram channels. Okay. Now, if you look at both the structures, what do you notice? You notice that here, it is just a multinucleated structure, no boundaries, no compartments. But here, if you see, you will notice that there are compartments, right? Students, there is septum. Okay, the presence of septum or walls which leads to the formation of compartments. Okay, compartments. But on the other hand, you have just, okay, just the nucleus. This will be just a multinucleated structure. Okay, just a multinucleated structure. No septums, no compartments. Fine? Yes, aseptate, we can use one more word, cenocytic. Okay, again, for the multinucleated, we say cenocytic. Cenocytic. That is clear? Okay, now example you have to remember, students. In aseptate, the only fellow that is going to fall here is Phycomycetes. Okay, Phycomycetes. Right? Phycomycetes. But what about septate, ma'am? You have Asco. Okay, Ascomycetes, Basidiomycetes, and Deuteromycetes. Deuteromycetes. All other fungi is going to fall under septate, but the only fellow who is going to fall there is Phycomycetes. Yes, good job, good job. Good job. Okay, now students, sometimes in the fungi, there is a stage, okay, there is a stage that comes where in your aseptate, okay, you will notice something like this, okay, you will notice something like this, that is, this is normal, yes ma'am, this is normal, but in one stage you will notice that there are two nucleus, okay, there are two nucleus, one green color I have drawn and let's say one yellow color I have drawn. This, can you see two dots here? Is it visible? Wait 1 second, I'll put red there, okay, red there. Now students, look here. There what is happening, here basically a dikaryon stage is formed. This one, what is happening? A dikaryon stage is formed, which I'll explain you again further. Dikaryon stage, students, it's an intermediate stage. Both the nucleus should have fused, because again in sexual reproduction you will study plasmogamy, that time karyogamy, when you study these two nucleus should have fused. But what happened? They came to an intermediate stage where both the nucleus are like that only. That is called a dikaryon stage. We can also call it dikaryo phase also, okay. Dikaryo phase, the small phase, no, like that. Okay, dikaryon, clear? Thank you, Sri Songs. Very sweet of you to tell that. Okay, very good. Okay, dikaryon stage. Okay, dikaryotic. All these words you can use. Dikaryotic phase, you can tell. Dikaryotic. That means it's an intermediate stage. It's an intermediate stage where both the nucleus are going to be there intact. They wouldn't have undergone fusion. Okay, undergone fusion. This is the whole about your hyphae. Okay, more than this slide beautification I cannot do, students. It's not just beautification, mainly it is for your purpose to study. Okay, and those who are posting any story, you can tag me on Instagram as well. Okay. Okay, yes. So this is your portion. Okay, now, oh yo. 1 second, students. Slide is running. Slide is like, why are you doing too fast? 1 second. Yes, reproduction. Now, very important thing about fungi is they do two types of reproduction, three types mainly. That is, one is going to be called as vegetative reproduction. Vegetative reproduction. Vegetative reproduction is going to happen because of budding, binary fission, and fragmentation. These are the three methods by which we can do vegetative reproduction. But it does not stop there, students. All the reproduction that you have studied so far will come here. Okay, will come here. Okay, that is going to be, yes, human physiology. I will be starting from next week. That means from Monday onwards, not Monday. Okay, so yes, you people can wait for human physiology revision. Asexual reproduction, students, happens because of three spores. Okay, first one is called as conidia or conidiospores, you can tell. Okay, conidia I'm writing. Okay, conidiospores. Sporangiospore, and zoospores. Asexual reproduction happens because of the help of spores, but sexual reproduction also happens. That is with the help of ascospores, basidiospores and oospores. So here the main thing about fungi is vegetative reproduction also is happening, asexual reproduction also is happening, sexual reproduction also is happening. Vegetative reproduction is because of budding, binary fission, fragmentation. Asexual reproduction is happening because of spores only, but conidiospores, zoospores, sporangiospores. In that, only conidiospores are exogenous. Exogenous means what, students? They are growing outside. Okay, exogenous. Can you see how they are growing? They are growing outside. Everything else is inside a sporangiophore. Always remember, whenever we speak about spore, remember the structure that I drew for the bread also, I drew one whole network or a thread-like structure, and then I drew a circle. Okay, I just drew dotted structure, but inside, remember, there's one circle. This will be called as a sporangiospore, where the spores are going to be there. Inside this, your spores are going to be filled. Inside this, your spores are going to be filled, and when the condition gets unfavorable, they will split open. Okay, they will split open. Okay, they will split open, and the spores will get scattered. They will get scattered. Clear? Clear? So, a structure will be there for the spores to be there, but then this fellow is standing outside. He's like, okay, here only you add on all the spores. So, conidia. Okay, conidia is exogenous. Here you can see sporangiospores, here you can see zoospores. Clear? But this is asexual reproduction. Anyone has any doubts? Anyone has any doubts? Yes, outside. Good, good. Yes. No doubts, right? Pakka? Good job, good job. Okay, good job. Yes. Now, students, sexual reproduction happens because of ascospores, basidiospores, oospores. In that, your basidiospores, okay, will grow exogenously. Exogenously. Outside this fellow will grow. Now, about spores, if you want to study, no, there are a lot of things. Aplanospores, that means the ones which will not be motile. Planospores, which are motile. Such kinds of, a lot about it is there, okay. Exogenous, aplanospores, are they motile, what is the shape of them, many things are there, but it's not there in NCERT, and it is, if you want to read about it, you can definitely read it on Google and other things, right? But there is nothing much about it. Like, they will not ask you how the conidiospores looks like. They will ask you, is conidia exogenous or endogenous? That's why I'm not going to get into the depth of that, okay? Depth of that. Now, here, reproduction, here you can see asexual spores are ascospores, basidiospores. Students, from the terminology itself, I want you people to decode something. What is it? Ma'am, ascospores will be part of ascomycetes. Yes or no? It will be part of this fungi, okay, and basidiospores will be part of that. Basidiospores will be part of basidiomycetes. Yes or no? Okay, basidiomycetes. So this you will understand. Now, deuteromycetes is does not have a sexual phase. That is why we are not talking about that, okay. Now we have one more which is called as the oospores, okay. You can see how it is happening, no, oogonium is there, oospores are there, antheridium is there. Again, here basidiospore, ascus, ascospores. So students, the structure where this ascospores is going to be there, it's called as ascus, okay, ascus. This one, can you see long structure? Inside this ascospore will be there. Same way, there is one structure called basidium. On the basidium, the acidiospores will grow. This much if you understand, it will be easy. Okay, it will be easy. Okay, next is sexual reproduction. Some important words are there. There are some events, okay? Events that will happen in your sexual reproduction. What are the events? All of you tell me in the chat box. All of you know. Events in sexual reproduction. Yes. Oh God, what is happening? Yeah, pakka means confirmed. Confirmed. That is what the meaning is. Okay, tell me quickly. Good job, good job. Who is that? Kubera, Meghna, Karthi, Roshni. Yes, correct answer, right? So here, events of reproduction. First you have what? First you have plasmogamy. Plasmogamy. Second you have karyogamy. karyogamy. Third, you have zygote formation. Zygote formation. Correct? Yes or no? Yes, ma'am. Yes, very good. Meiosis or we tell. So here what is going to happen, students? Plasmogamy, something to do with your protoplasm here, okay? So fusion of protoplasm leads to plasmogamy. Protoplasm will fuse. That means cytoplasm plus the nucleus, protoplasm will fuse. Second is going to be fusion of the nucleus. Fusion of nucleus. Last one is zygote formation. This is going to happen with the help of meiosis leading to the formation of zygote. Now what happens here? After your protoplasm fusion, that is when your dikaryon stage is going to be formed, right? So as I told you, students, dikaryon stage is going to happen. Let's say this is a parent. Okay, I'm drawing not a fungi structure, but this is just to make you understand. Okay, this is a parent. This one has a nucleus. Can you see a big yellow color nucleus? Okay, yellow is visible. Very good. Now we have one more. Okay, one more parent who's blue color parent. Okay, this is one more parent. Okay, so let's say this is parent 1, this is parent 2. Now protoplasm will fuse. Yes, ma'am, protoplasm will fuse. So what I will get? I will get one cell, okay, where the protoplasm fused. But in the next stage, nucleus should have fused. Nucleus should have fused. But what happened? Here what is going to happen, students? One nucleus from the parent 1 and one nucleus from parent 2 will remain unfused. Okay, will remain unfused. Two nuclei from different parents will share a single cytoplasm. Will share a single cytoplasm. Got it? This is what is dikaryon stage. This is important for you. Okay, dikaryon. Now students, this is seen. Okay, this is seen in Ascomycota and Basidiomycota. That is again seen in your Ascomycetes and Basidiomycetes. Clear? This is present. This is shown in Ascomycetes and Basidiomycetes. So two nucleus from different parents will share a single cytoplasm. No doubts in this, I hope. Structure is perfectly clear. Yes. After the formation or the fusion of the nucleus, what will happen? Zygote formation will happen. Same thing happens in us also. Two gametes will come, fertilization will happen, zygote formation will happen. Same thing here also. Zygote formation will happen. Okay? Yes. Very good. So this dikaryon stage, please understand, this is like an intermediate stage. Okay? We can call it like an intermediate stage because usually, okay, usually fusion should have happened. Normal situation, what should have happened? Fusion should have happened. Zygote formation should have happened, but that did not happen. Instead, two nucleus stayed separately. Two nucleus stayed separately. Very good. Yes, good job. Okay. Yes, very good. So later, what is going to happen, students? Here, haploid spores will be formed after meiosis happen. Haploid spores will be formed. Yes, this is your dikaryon stage and fusion. Everything is covered, no? Plasmogamy, karyogamy, zygote formation. Okay, events also done. Events also are done. We will just remove this. We don't need this. Okay, one more topic which is not like in the sense sometimes when we teach, some people overlook that, okay, students already know it, but this is one more thing important, students. Gametes, depending on their shape and their motility, okay, if they are aplanospores or planospores, their motility, they will be categorized into three categories. Okay, what are the three categories? You guys already know it. Okay, depending on their what, students? size and mortality, they will be categorized into 3 categories. Okay, 3 types of gametes. First I'll draw the structure, then I will explain the structure for all of you. Okay, yes, very good. Who is that? Kubera, very good. So here what is going to happen, students? First category is going to be called as isogamous. Okay, isogamous. What is isogamous? Look here, all of you. I will draw the structure and show you. They can be motile, they can be non-motile. It is up to them. Okay, but I am drawing motile. Here, look here. You don't tell me it looks different, ma'am. It is same. Okay, that means here these people show same size. And even motility is going to be same. Okay, these people are isogamous. Iso word, from that only you will understand, no? Next one is anisogamous. Anisogamous. Here, what is going to happen? One is going to be bigger but motile. Okay, one is going to be smaller but motile. That means just difference in their size. Okay, just difference in their size. Different size. Got it? Last one is oogamous. Okay, oogamous. Just like in us. Okay, humans. What is going to happen in humans? A big female is going to be there, which is not going to be motile, and then a small sperm will be there, which is motile. No, same thing here also. One is motile, one is non-motile. Okay, one is motile, one is non-motile. So here they have both difference in size and motility. Okay, these are the 2 things. Fine. So 3 types of gametes are there: isogamous, anisogamous, and oogamous. Perfect. No doubts in this, because so many times this terminology is repeated in your NCERT. There might be some students who don't know this. So just make a note of this. Okay. Make a note of this. Now we will go to kingdom fungi. So students, kingdom fungi, most of it I have put NCERT, because when I write, some words and points can be, by mistake, in case it skips. So we are going to read NCERT and I'm going to make you understand. Okay. Now kingdom fungi, how do you think the classification happened? Ma'am, classification would have happened on morphology. Classification could have happened on spore formation. What else? All of you, those who are revising this chapter, tell me in the chat box. What else could be the reason for fungi classification? Hyphae, mycelium. Jaldi you should answer. Okay, jaldi if you answer only, we will be able to go forward. Yes or no? Very good. Spore formation I wrote. Morphology I wrote. Is morphology correct? It's one line of your NCERT. I want all of you to check that line of NCERT and tell me. Is it mycelium? Is it hyphae? Is it spore formation? On what basis did they do the classification? It's easy. You can tell. Very good. Who is that? Meghana. Very good. Fruiting bodies. Based on fruiting bodies. Perfect. Excellent. Okay. Good job. Yes, fruiting bodies. Okay, here students, see, Phycomycetes, Ascomycetes, Basidiomycetes, and your Deuteromycetes. These are the classes. Okay, first we will start with Phycomycetes. What are the important characteristics of Phycomycetes? Look here. Members of Phycomycetes are found in aquatic habitats, on decaying woods, in moist and damp places, or as obligate parasites on plants. Okay, as I told you, these are general characteristics of fungi. They will be present in all the moist conditions. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospores, which are motile, or by aplanospores, which are non-motile. That means aplanospores means non-motile, which can, or zoospores, which are motile. These spores are endogenously produced in the sporangium. Endogenously means inside. So they are telling that it is formed inside like this. Okay, it is formed inside like this. The spores are endogenously present. Okay, the spores are endogenously produced in the sporangium. Sporangium. Got it? All of you? Good job. Now, a zygospore is formed by the fusion of two gametes. These gametes are similar in morphology or they could be dissimilar. That means they can be isogamous or they can be oogamous or they can be anisogamous. Okay, no difference here. They can be any of this. Now, some common examples are Mucor, Rhizopus and Albugo. So, students, here the example you are going to remember, since Phycomycetes is the first person, his name is going to be Ram. What is his name going to be? Ram. Okay, R for Rhizopus. Okay, Rhizopus. A for Albugo. And M for Mucor. Okay, these are going to be Phycomycetes. What is the most, what do you have to remember about them? The first or most important thing is they are aseptate. Okay, they are aseptate. Yes, got it? Asexual reproduction happens with the help of zoospores. Okay, zoospores. Okay, a zygospore is formed by the fusion of gametes here. That means here also all the three stages happens. Okay, yes, Ram. Very good, very good. So, Ram is the first son's name. Next one. Mycelium is branched and septate. This is going to be Ascomycetes. Okay, mycelium is branched and septate, right? This is going to be Ascomycetes. Okay, Ascomycetes. Branched and septate means like this, the septate will be there. Like this, branching will be there. Okay, branching will be there. It's not necessary all of them should have branching, but these people have branching. They are branched and septate. The asexual spores are conidia. What is it? Asexual spores are conidia which are produced exogenously. That means here they are telling that conidia is going to be present like this. Okay, like this structure, conidia is going to be present. This conidia is going to grow outside like this. Exogenously, it is going to be present. Any doubts in this? Exogenously, it will be present. Very good. Exogenously present. Okay, exogenously present. Conidia on germination produce mycelium. Sexual spores are called as ascospores. This I already told you, which are produced endogenously inside a sac. This also I told you. So since we already did the introduction, it will be more easier for you. So I told you, students, these are formed inside, okay, a structure like this, inside which you have the ascus. Okay, ascospores are there. The inside the sac-like structure called ascus. See, understood? Singular it is asci, or you can write ascus. So this is the ascus, and this is your ascospores. Because every spore should have a structure, because they have to be protected until the condition gets favorable. So for that protection, everything will be packed. So this fungi can also be called as sac-like fungi. What can they also be called as? Sac-like fungi, because of this term. Okay, they are also called as sac fungi. Okay, this Ascomycetes is also called as sac fungi because of this. Perfect. Any doubts? Okay, this you have to read NCERT, but I am just explaining you the terminologies. Okay, now these asci are arranged in different types of fruiting bodies called ascocarp. Some examples are Aspergillus, Caliceps and Neurospora. What is the name of this guy? His name is Can. Okay, so the second son's name is, first son's name is Ram, so second son's name is Can. Okay, C for A for Aspergillus, N for Neurospora, which is used for many other genetic, many other genetic work. See, Neurospora is used extensively for genetic works and in biochemical industries. Okay, biochemical industries could be asked as an MCQ. Okay, could be asked. Now, many members like murals and truffles are edible and considered delicacies. Students, this is used in our ramen. All of you watch your anime and all. in that ramen they use this murals and truffles which are edible, okay, edible fungi, okay, edible fungi. Now moving forward, next one is Basidiomycetes. Okay, Basidiomycetes. The mycelium is branched and septate. So I told you already, in septate itself I told you Ascomycetes and Basidiomycetes. So now we are just going like an overview because the introduction was clear. I already mentioned everything in the introduction, so this is going to be easier for you. Mycelium is branched and septate. Okay, mycelium is branched and septate. The asexual spores are generally not found, but very important here, asexual spores are generally not found. That means there we had zoospore and conidia, but here we do not have any asexual spore. No conidia, no zoospores, no sporangiospores. Okay, but reproduction by fragmentation. That means vegetative reproduction is common by fragmentation. The sex organs are also absent. So asexual reproduction also is not there, sexual reproduction also is not there. But what will happen? The somatic cells, that means the plasmogamy is brought about by the fusion of two vegetative or somatic cells of different strains or genotypes. So what we will do? We are going to bring about this by taking two different somatic cells. Okay, let's say this is somatic cell 1. Okay, they are of different genotype. Then we have one more somatic cell, again another genotype or a different strain. So I am writing it here, strain 1, okay, strain 2. Like how you had your rough strain and smooth strain, like the different strains will be taken. These two strains both are somatic, okay, both the cells are somatic because they do not have sex organ, that means no gametes are there. So somatic cells are there. These two are going to end up forming your, that is, they are going to do your plasmogamy and all of that, no? So these resultant structure is dikaryotic, which ultimately give rise to basidium. So these two will ultimately, okay, they will form like this, and they will have one red nucleus and one pink nucleus. They'll have one red nucleus and pink nucleus. Why? Because, ma'am, these people are going to form your dikaryon stage. Dikaryon stage. And this dikaryotic stage or this dikaryotic phase will ultimately mature and give rise to your basidium, which I showed you before. This is the basidium. Anyone has a doubt in this? This will give rise to the basidium. This will give rise to the basidium. So what happened, students? We took two somatic cells, both of different strains or genotypes, okay, and those both will do plasmogamy. Okay, plasmogamy will happen. That means basically fusion will happen of the cytoplasm or the protoplasm, giving a dikaryon stage where both the nuclei of the different strains will stay in a single cytoplasm, and they will end up forming a basidium. Okay, they will end up forming a basidium. Okay, now that basidium, remember, karyogamy and meiosis takes place in the basidium, producing 4 basidiospores. So again, what happens, students? We formed basidium. Basidium was formed from where? Ma'am, from the dikaryon stage. Okay, this was the stage. Okay, one red nucleus we had from one strain, and we had one pink nucleus from another strain. These two ended up forming the basidium. This is the basidium. Now this basidium, in the basidium only, two more things are going to happen. What? Karyogamy plus meiosis is going to happen, leading to this basidium. Okay, this basidium forming basidiospores. Any doubts? Clear? With the structure I have taught you, okay, they are exo. Yes. So, see students, exogenously produced on the basidium. The basidia are arranged, okay, single individual bodies. No, basidia are arranged in fruiting bodies called basidiocarp. So, there also we had ascocarp and we had basidiocarp. Okay, many of them arranged together to form your basidiocarp. Clear? Yes. Rashmi Sahitya, I do not see it related to the topic. So, when I teach you biotechnology, you can ask the question. Okay? Got it? Okay. No doubts, right? Okay, perfect. Perfect. Okay, okay. So students, remember all of this is going to be protected. Okay, that is your basidiocarp. So carp means they are the fruiting bodies. Always remember, please make a note somewhere and keep, because you will get confusion when you are studying by yourself. Write that this basidiocarp, ascocarp, they are all fruiting bodies. They are the fruiting bodies of this fungi. Okay, inside the fruiting bodies only all these spores, everything will be present. Okay, yes. Now common example is going to be Agaricus, Ustiligo and Puccinia. What is the name of the last fellow? So we had Ram, we had Can. Now we have to make a name. No, what is the name last time I made? A-U-P. U-A-P. P-A-U. Po. What did I make, students, last time name? Ube does not make sense. Okay. Yes, tell me. Okay, A-U-P. So you have first Agaricus. Okay, Agaricus. Ustiligo. That is again smut, okay? And Puccinia, which I told you in the starting, wheat rust. Okay, red color rust that forms. Okay, got it? Yes, perfect. Now, Ram can opt. Okay, Ram can opt, no, like that, opt, A-U-P, opt for cat. Cat is the example for Deuteromycetes. So you can write Ram can opt for cat. Yes, perfect. Very good. Now, students, Deuteromycetes, don't faint looking at this big paragraph which is bigger than me. I will just tell you what you have to study. First of all, understand Deuteromycetes is an imperfect fungi because of the fact that their sexual phase is not known. Okay, and when their sexual phase was known, they were moved to Basidiomycetes and your Ascomycetes because they were not belonging to Deuteromycetes. So a lot of rearrangement has happened in Deuteromycetes, and because of which these people are called as imperfect fungi. Okay. Now look here, it is also possible that the asexual and the vegetative phase have been given one name and placed under Deuteromycetes, and sexual stage another name is given and they are placed under Ascomycetes or Basidiomycetes. Understand, students? Perfect stages of the members of Deuteromycetes were discovered and they were often moved to Asco or Basidiomycetes. Fine. The Deuteromycetes reproduce only by asexual spores known as conidia. So who are the two people that are producing conidia? Tell me in the chat box. Who are the two people who are producing, that is, asexual spores with the help of conidia? These kind of questions only will come for you people. Okay, the mycelium is septate and branch. This also I discussed with you already. Septate and branch. Some members are saprophytic or parasitic. Okay, while a large number of them are decomposers. Very important. Decomposers of litters and help in mineral recycling. So they are not useless, just that their sexual phase is not known, but their asexual phase is known with the help of conidia. Okay, conidia. Now tell me the answer for it. Tell me the answer for which are the ones that are going to reproduce by conidia in the chat box. Tell me. Battery is low. Run to the charger. Go sit near the charger quickly. Yes, it is going to be Answer is going to be Ascomycetes and your Deuteromycetes, right? Very good. Ascomycetes and Deuteromycetes. So students, Deuteromycetes do not have a sexual phase, because of which they are called as imperfect fungi, but they do have an asexual phase by the help of conidia. Even though their sexual phases were known or tried on understanding, many times they were shifted to Ascomycetes or Basidiomycetes, or their names were given under them. Okay, here you should understand that these people are maximum decomposers of litters and they help in mineral recycling. Now students, some examples are Alternaria, 𝐂𝐡𝐞𝐤 and Trichoderma. Trichoderma is pathogenic because we know it causes ringworm allergy. Okay, ringworm allergy. So here the example to remember is cat. Okay, cat. Okay, 𝐂𝐡𝐞𝐤. A for Alternaria. Alternaria, you can remember, like people who keep altering, no? So they are like very imperfect people. Okay, Alternaria and Trichoderma. See, almost all chapters are connected to each other. See, Trichoderma you have studied there also, here also. Perfect. Any doubts in this? Anyone has a doubt? Will you do 2-3 questions now? Good job. Okay, first question on the board. Which of the following organisms are known as chief producers in the ocean? NEET 2018 question, and this will be your score for GM. Okay, rest of your score will come from this. See, in the description box, biological classification and living world is there. I want all of you to solve and drop the screenshot on my Instagram. Okay, or put it in the comment section that ma'am, I scored this much. GM Express, I scored this much. Okay, GM Express, I scored this much. Epidermophyton, microsporum. Yes, cyclosporin A, no? Okay. Yes, tell me the answer. Very good. Good job. The answer is going to be diatom. So +4 in your wallet. Okay. Next question. Which of the following is a correct statement? Cyanobacteria are a group of autotrophic organisms classified under kingdom Monera. Bacteria are exclusively heterotrophic organism. Slime molds are saprophytic organism classified under kingdom Monera. Mycoplasma have DNA, ribosome and cell wall. Which of the following statements are correct statement? That means only one statement will be correct. 2022 question. Yes, 2022 question. Yes. One. Yes, what is the answer? Cyanobacteria are a group of autotrophic organisms classified under kingdom Monera. Bacteria are exclusively no. Slime molds are no. Mycoplasma have no cell wall. Right? So here the answer is going to be option number A. Very good. +8 marks. Those of you got 8 marks, put it in the chat box. Methanogens belong to Archaebacteria, dinoflagellates, slime molds, U-bacteria. 2016 second question. Okay, that Manipur paper, not second, part 2 question. Ouch. Yes, fast answer and put your scores as well. Okay, put your scores as well. And do like the video. Don't run away before liking. Some of you will answer and run away. Yes, good job. The answer is going to be Archaebacteria. +12 in your wallet. Okay, +12. Next question. In 5 kingdom classification, I think it is not visible. I'll read it for you. In 5 kingdom classification, which single kingdom contains blue, green algae, nitrogen fixing bacteria and methanogenic archaebacteria? Monera, Protista, Plantae, Fungi. In 5 kingdom classification, which of all this is present in which one? Is it in Monera, Fungi, Protista or Plantae? Yes. Tell. Very good. It is Monera. Easy peasy question it is. Plus 16. Okay, plus 16. Next question. Fungi usually store their reserve food material in the form of. I forgot to tell this, so now this question I will answer. Okay. Fungi usually stores, but I have taught you people before and you should know NCERT. So tell me the answer for this. After this, we have viroids, virus and lichens. After this chapter will be done. Okay. But your question is still pending. You have to do the question after the class ends also. Yes. Tell me the answer for this as well. No. Think properly. Open your NCERT and see the answer and tell me. Don't tell Chuma. Looking at other people's answer, they might be wrong. Okay, they also might not know just like you. Yes, good job. It's glycogen. Okay, and oil is given. Here it is glycogen. Okay, glycogen is the answer. That is how fungi will store their reserve food material in the form of glycogen. It is important. Okay, it is important. Remember and keep. Okay, I will write it in the notes as well. So this will give you plus 20. As usual, my plus 20 is done. Okay, rest you will do it from the NCERT. Now students, kingdom, Plantae and Animalia, I am not going into this because plant kingdom will be taught by sir. Alternation of generation, one word only is given there, and animal kingdom I have only taught. Okay, so we are not going into that 2 paragraph. Now we will go to virus. Okay, very important. Now see, all of us would have gone through a cold or a flu, that is cold, which is caused by rhinovirus. Now we spoke about HMPV virus. So like that, virus keeps coming and going. But what makes virus so exceptional compared to a bacteria or a protozoan or a helminth or something else? It is because of the fact that these people are not completely living, not completely non-living. The moment they come in cross with a host, they will release their genetic material inside and manipulate the genetic material or change the genetic material of the host. Right, that is the reason why HIV is so dangerous. Now look here students, virus, since they are not considered truly living, because still debates have been happening about understanding if they are actually living or non-living. So this question will keep going, right? Now let's say one person will tell ma'am they are non-living, one person will tell they are living, because they do have some characteristics of living, some characteristics of non-living. Okay, now the virus are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. So they have an inert crystalline structure. Now once they infect a cell, what do students? They take over the machinery. This is why they become dangerous, because these people will come inside, they will change the machinery inside our body, telling that hey, don't make host DNA, make my DNA. Hey, don't make host protein, make my protein. So this fellow will manipulate or completely change the machinery of the host. So even our body will not be able to recognize like this. This will keep increasing and increasing. Okay, now would you call virus living or non-living? Virus means venom or poisonous fluid. Now Ivanoski recognized certain, in 1892, recognized certain microbes as casual organisms of mosaic disease of tobacco. These were found to be smaller than bacteria, because they were able to cross through that. Students, when we make tea and all, have you seen? We put tea leaves, we put cardamom, okay, cardamom, yeah, elachi and all that, no? Now, if I am taking a sieve and I am filtering it out, cardamom will come, tea leaves will come out. That means the size is bigger. than the sieve holes. Here also we took a bacterial filter and we tried it, and we noticed this virus, they are smaller than bacteria. So they were like, okay, wait, they actually, Ivanoski thought that there were some casual organisms, or there could be a bacteria that caused this mosaic disease in tobacco. But later they realized these people are so small, they're smaller than bacteria, that they're coming out of the sieve. Now they passed through the bacterial proof, okay, filters. Then Bejnik, M.W. Bejnik in 1898 demonstrated that extract of infected plant of tobacco could cause infection in healthy plant. Could cause infection in healthy plant, and named a few new pathogen called as virus, and called the fluid as Contagium vivium fluidum, that means infectious living fluid. So he took the fluid from the tobacco, and he noticed that, oh my god, this fluid is enough to infect an healthy plant. So he called that fluid as infectious living fluid, that is Contagium vivium fluid, and he called that organism as virus. Now Stanley in 1935 showed that virus could be crystallized, and crystals consist largely of proteins. So he told that the structure of virus is not as simple as we thought. These people can become obligate parasites. And how can they become obligate parasites, students? Also because of their presence of protein and this DNA genetic material. So according to Stanley, he noticed that when I am trying to crystalline this, I am getting something, I am getting more of a protein. Do I have a shape like this? No. Yes, look here students, I have drawn a very big structure. Okay, now this is the protein portion. Okay, now here let's say I'm drawing like this. Something like this. Okay, something like this. Okay, yes, so here what is going to happen? Okay, like this. Now here, if you see here, the outside coat, okay, yes, Tanuja, okay, this outside coat is a protein. It's made up of protein. And what is this protein made up of? They are made up of small units called capsomeres. Many capsomeres together forms capsid. And this capsid is going to protect the genetic material inside. It could be a single strand, okay? It could be a single strand RNA, or it could be a double strand DNA, right? It could be any one of this, single strand or double strand. Single strand or double strand. It could be an ssRNA, or it could be a DNA. Depending on the host it is going to affect, depending on the virus, we have different types of it. So this is the structure, and they notice that this protein is made up of capsomeres, and together we can call it the capsid. Okay, no doubts in this, I hope. Now, moving further, in addition to the proteins, virus also contains genetic material that could be RNA or DNA. No virus contains both RNA and DNA, but there is an exception. Very recently, I read about it only, which was that leucovirus, if I'm not wrong. Okay, there is a virus which has both RNA and DNA. You people can check and tell me in the chat box. A virus is a nucleoprotein and a genetic material is infectious. In general, virus that infects plants have single strand. That's why I told you, depending on the host, this fellow will keep changing its genetic material. If it's infecting plant, it will go for that. If it's infecting host, animals like us, it will do DNA. Or if it is not able to do any of this, what it will do? It will take the help of retrovirus and it will convert DNA into RNA, RNA into DNA. Correct? Now, they'll neither have single or double-stranded RNA or double-stranded DNA. Bacterial virus or bacteriophage virus that infects bacteria are usually double-stranded. DNA virus. The protein coat called capsid made up of small subunits called capsomeres. I just told you, why they are present, students, so that they can protect the genetic material. They can protect the nucleic acid. Easy, no? Now, these capsomeres are arranged in helical or polyhedral geometrical forms. Okay, like what I just drew, no? If you look here, what I just drew, if I draw it this way, if they're like this sleeping way, then it will be like this. Given in your NCERT, no? Like this. Polyhedral helical structure, they will look like this. Like this. Like this, something like this, okay? I have drawn it like a peacock, but then it is a more, okay, hexagonal structure. Okay, like this it will look. Fine? Yes or no? Okay. Now, AIDS in humans is also caused by a virus. Which is the virus? Human immunodeficiency virus, HIV virus, right? We have studied everything about HIV virus. In plants, the symptoms can be mosaic formation, leaf rolling, curling, yellowing and vein clearing, dwarfing or stunt growth. So, if a virus and if infects a plant, no, very dangerous, okay, very dangerous. What is going to happen? Okay, what is going to happen is so dangerous, students, that leaf curling will happen, yellowing will happen, vein clearing will happen, like that many things will happen, okay, if it is affecting a plant. Now, students, this is the most important portion here about virus, viroids and everything is given here. One paragraph you have to study. In 1971, D.O. Dinner discovered a new infectious agent that was smaller than virus that caused, okay, it was smaller than virus, very important. It caused the potato spindle tuber disease. It was found to be free RNA. That means this fellow did not have protein. He was a free RNA. Free RNA ones are called viroids. It was smaller than virus. Okay. Then the RNA of viroid was of lower molecular weight. Prions are what then? In modern medicine, certain infectious neurological diseases were found, like your mad cow disease. They noticed that this fellow is just free protein. He's just the protein. No genetic material. This fellow was called prions, okay? Remember like this, P protein, okay? P protein. This fellow ended up causing your mad cow disease, okay? Also bovine spongiform encephalopathy, okay? Encephalopathy. They might ask you the full form of BSc, so please make a note of this and keep, okay? Please make a note of these. These agents were called prions. The notable diseases caused by prions are BSc, commonly known as mad cow disease, and also in humans it caused the Jacob disease. Again, CR Jacob, it has a full form, okay? Yes, now lichens, already I told you, students, it's a symbiotic relationship between fungi and algae. Now fungi has a name and algae has a name. Fungi, that portion of the lichen is called as mycobiont, and algae it is called as phycobiont. Just remember like this, photosynthesis, phycobiont, okay, phycobiont. Now mycobiont means the fungal component, okay. These are the two components of your lichens, mycobiont and phycobiont, and which are autotrophic. So what are they going to do for each other? They are going to do something like this. Okay, wait. I'll show you. This is your something like this your lichens will look. Okay, it will be white outside. So the white portion is their fungi, which is also called as your mycobiont. Okay, and the middle portion will be your algae. Okay, the middle portion will be your algae. And what is algae going to do for the fungi? Algae is going to provide food for the fungi, and fungi is going to provide protection for algae. Okay, algae is going to provide food, fungi is going to provide protection. Clear or not? That is your phycobiont and mycobiont, that is your lichen. And their association is symbiotic relationship because both are helping in each other. One is providing shelter and one is absorbing nutrients and water for the partner. It is not like harming each other, both are helping each other, both are in full happiness, okay, full happiness. Clear? Chapter is over. Chapter is over. Now chapter is over means don't run home. What you will do, first of all, you will like the video, then you will click on this, okay, right, enter your phone number or you continue with Google. Okay, now let's say I continue with Google. Okay, again I have to enter my phone number and put my OTP. Okay, after that you will be able to get the test. Okay, entire test will be available for you and you can solve the test and let me know in the marks, let me know the marks in the comments. Now students, for those of you who are looking for a 3 months crash course, I have made a video here. You can go and check out what and all is available for you at this price range. This is the video. Okay, it is not 37 minutes, it's some error. It is only an 18 minutes video and you will be able to know exactly what you're getting for the price that it is mentioned. Clear? So the combination is going to be 50% live classes and 50% recorded. So the price is reduced. Okay, drastically the price is reduced. For 3,500 you will get the batch live plus recorded classes. You will also get pedagogy, you will get access to all the books. At the same time you will get all India test series at your home. Okay, you will get it delivered at your home. So for this price at this time you will not get a batch where you will have all India test series. For all India test series itself people charge you 2,000 to 3,000 rupees. So instead of that, take this batch. You will have all the one shot lectures, you will have repository of all the topics. Okay, and at the same time you will also get, and this recorded videos you can watch it at your pace, 2x, 1.5, 1x, 2x, whichever speed you want to watch you can be watching. Okay, yes. So those who attended the first class, please let me know in the comments how the class was, and those who attended the last class of Biology 360 Club. And Biology 360 Club is very close to my heart. Why? Because this name we curated it over, overnight we curated the name, and also we were able to deliver the highest weightage classes. Okay, highest weightage classes. So most of the topics, that is, we have done 60 marks. If I want, I will go back to the channel. Okay, just look here, and we are 186k today students, so which is also a very happy news. Those who haven't subscribed, quickly subscribe, so we will become 200k. See, you can see we have done 40 marks. Okay, biotechnology 60 marks, genetics, and we have done again 25 marks. Okay, nothing, we have not written anything fake. Everything is according to your previous years. 25 marks we did here, and 40 marks we have done reproduction unit. So we have done a range of around 165 marks in Biology 360 Club, including today we will have a overview of maybe 170 marks. Okay, so yes. You people are teasing, okay. You people suggest a name. So now we will start with your backlog series students, where we will go back to your chapter 11th, okay, class 11th. Now today we have already finished one portion of it. Living world and biological classification is done. Now I will start with human physiology, and I'm mostly expecting to start from body fluids and circulation because I've not done a revision for you for that chapter. I have done Tejas series. I have also done for you question practice on that chapter, but I've not done a full-fledged revision, so I'll be doing that. Okay, if you're excited, please let me know in the chat box if you're excited for backlog series and how you felt Biology 360 Club was, because I really want to know your feedbacks. Because if your feedbacks are really good, we'll be bringing back Biology 360 Club, but in separate ways. Okay, because I cannot mix plant physiology, human physiology together, so because of that we'll be bringing it separately. Okay, yes, from Monday we will be starting. I think Monday you will not have a class, but Tuesday and Wednesday you will have a class. So thank you so much for joining students. See you all in next class, and I hope all of you enjoyed Biology 360 Club. I truly enjoyed this club. I truly loved taking long lectures and making notes and everything. And don't worry about anything. Don't worry about the notes. This beautiful notes are only made for you people, and the notes will be available immediately after the session in the description box. I will share the link as well. All this I have written and made for you people. So this notes will be available in the description box for you. Okay. Yes, happy Pongal to all of you who are celebrating. I think Monday some of you will be celebrating, and some of you on Tuesday if I'm not wrong. So happy Pongal, stay healthy, stay happy, have a good time with your family, but at the same time don't forget about your aim, don't forget about your goal, because that is going to define your future. That is going to be a mark in your future. If you do it well, everyone will appreciate you. If you don't do it well, everyone is going to pull you down. So you should decide your future, and you should decide what is next for you. So see you all in next class. Till then, thank you so much. Okay. Yes, do not forget to let me know in the comments how Biology 360 Club was, because it's very close to my heart. At the same time, I would like to know your feedback so we can come up with more sessions for you. Everything is for you students. For us, it is we have to take a class, we will take a class. The banner name does not matter until it is meaning something for you. Okay, so see you all, bye bye.